up. Okay, so we're ready. So I want you guys to um you guys see my next slide, right? You guys see this? All right. So before we begin, everybody has their phones and everybody is connected to the internet, I suppose. Is it correct? Am I correct in that assumption? Right? Everybody has devices in front of them. Hannah, you have yes, before we begin, all right, I'd like to ask everybody. I'm gonna give everybody a couple of 5 minutes. Okay, I'm gonna give everybody 5 minutes to post something in their social media accounts, whether it's on Twitter, whether it's on Instagram, anything about this one. You can take a picture of this one, you can take a screenshot of this one. I'm gonna give you guys 5 minutes, and then just post something in Instagram or anything in your story or anything in your Facebook story or anything. You can even post it on Twitter or whatever, um, about blood donation, because I'll tell you guys later why I asked this. Okay, after your 5 minutes. Okay, if you don't mind, it's not what they call this. It's not a mandatory request, but yeah. All right, so a moment of silence for our donors also, and for the people who have died requiring blood samples, requiring blood bags. So yeah, let's try putting 3 minutes only, not 5 minutes. I'm already late as it is. All right, so 1 pint of blood can save 3 lives. Every blood donor is a lifesaver. Give blood and make the world a healthier place. Blood can give someone a chance at life. So anything related to blood donation, please help me out and try to post something in you on your social media accounts, because you guys are one of the luckiest people in the past 100 years who have relatively the easiest access to information. And I think out of 47 people, we could touch lives. All right, four, 47 people here, one of the 47 people here can touch someone's life and ask them to involuntarily ask them to send to maybe ask a person to in your in your vicinity or in your group of friends to donate a blood donate his or her blood. All right, so okay, so my timer is up. Hopefully people have posted something in their social media accounts about blood donation. Anything, you can just take a picture of this one, or you can copy and paste some of the quotations that I put there for you. Now, we got to begin with our discussion with history, because in the board exams, even in the past, they've always asked the history of blood banking for everybody's information. Okay, so it may not be asked in blood bank, but it may be in your blood banking board exam. But it may be a question in the in medtech laws, which medtech laws and the histopathology portion of your medtech laws, or MTL, and bioethics slash histopathology, which is actually the last test in the Philippine board exams. So I'm going to run through some of the most important or the commonly asked questions in the history of blood banking. So we're gonna start first with the father of modern blood banking. It's actually a misnomer to put Karl Landsteiner as the father of modern blood banking or blood banking in general, because he wasn't the person who invented blood banking techniques. It was actually, or pioneered rather, not invented, pioneered the development of blood banking techniques. And it's actually the first chairman, or rather the first president of the American Red Cross. It's Charles Drew, Dr. Charles Drew. He's here. That's the picture. Photo credits are not mine. It is from the American Association of American Medical Association, And some, I think I have another one on the next slide, but no, no. Okay, so that's one question that you're going to get, you might encounter in your board exams. That's why I added it here. Now, another person of of paramount importance that you guys need to remember is the first chairman of the Philippine Red Cross. Um, Anybody has an idea who's the president, who was the president when the Philippine Red Cross was established? It was actually what? It was actually who? Anybody in the class who is familiar with the first Philippine Red Cross? Who's the Philippines when the Philippine Red Cross was established? It was actually the late President Manuel L. Quezon. And his wife is actually Aurora Aragon, who was the first chairman of the Philippine Red Cross. Nowadays, who is the chairman of the Red Cross? Can anybody answer? His name has been coming up on the news lately, primarily because of the bi- who? Who, Hannah? Yes, that is correct. I think somebody here is updated with the current events. And yes, Hannah, that is correct. It's Richard Gordon right now. All right. Now, photos are not minus, I guess, and that's from a photo that I just grabbed in the Red Cross, the Philippine Red Cross. Now, he's the first person to discover. Who was the first person to discover ABO blood groups? Now, this is an easy question. Hopefully, somebody can unmute themselves. Hopefully, one of you guys can unmute yourself and then tell me what's the answer to this particular slide. Anybody can answer? All right, a lot of people have been able to answer the question correctly. Wonderful. This is his picture. He looks very anxious in that picture, but that is a picture of Karl Landsteiner, which was grabbed from wikipedia.org. No, sorry, it was in thoughtco.com. All right. Now, who discovered ABO blood group system? It's actually two people. It's Sturley and Von Di Castello. Unfortunately, I wasn't able to grab a picture of him on any other source that I have, but but these are the two people the who had discovered the AB blood group. So the discovery of ABO blood groups is actually from Karl Landsteiner, and the AB blood group was discovered much later, probably around 5 years, 1907, if I'm not mistaken. 1907. All right. Now, genetics. We have to review genetics because you guys are going to study you need to learn some general terminologies, as well as understand some of the concepts related to genetics, that which will be applied in blood banking. Now, I have a question. Is anybody here familiar with the Punnett square? Anybody is familiar with the Punnett square? I'm gonna open something. All right, so I think some people are commenting. You can comment on the chat box. Don't worry, I wouldn't mind. Are you familiar with the Punnett Square? Anybody in the chat? You can share your thoughts with us. Yes, po, sir. Angela Felipe said yes, po. Right. Wonderful, wonderful. So wait, let me show you a bit of a drawing for an illustration for you guys. All right. I'll stop this. I'm gonna stop the presentation here for a while. Let me delay your screen for a while now. I'm gonna share my entire screen. Can you guys see my entire screen? Can you guys see it? All right. So, okay, wonderful. All right. Let me just enlarge this one and get a thicker brush, or rather a. Okay. All right. So this is I'm gonna put a box for a while there. Okay. So Punnett squares, because you will be asked questions in your board exam. What is the percentage of the likelihood, or what is the likelihood of the offspring of two parents' blood group? Now, what is your blood type? What is your blood type, Hannah? Can you please tell me in the chat what's your blood type? So O positive. So let's try with the O. We're staying first with we're gonna stick with O. So what are the genotypes that may be associated with O? O positive. It's either AO. Sorry, it's OO only. All right. What about Zyrene Alvarez? Because her name is quite rare. Let's try. What's your blood group, Zyrene? O positive. No, let's find another person. What about Justin? What is your blood type? Justin Javier, can you tell me what's your blood type? Ay, this one is wonderful. So B positive can have two types of genotypes. All right, can have the possibility of having two genotypes. All right, so let's talk about the genotypes of Justin's. Hello? Can you guys hear me? Oh, really? I'm getting disconnected all of a sudden. I don't know why. All right. Is it okay now? Can you hear me now? Can you hear me now? All right, so we're gonna select, for example, let's assume that Hannah and Justin got married, and the genotype of Hannah is OO, obviously because of codominance. Obviously because of codominance. Nothing too serious here. It's all theoretical. Okay, so Hannah is OO, her genotype is OO. And let's assume that we did molecular studies for Justin, and it was found out that he is BO. His genotype is BO. Now, how many percent what's the percentage of Justin Justin and Hannah's offspring? What's the percent? What are the possible? Well, let's start first with the possible blood groups of their offspring. Can anybody tell me in the chat what are the possible blood groups of Hannah and Justin's offspring would be? Possible blood groups. We're not asking for 50% first. Chill ka lang, Rina. Rina, chill ka lang, ha? We're not asking for B and O are the possible. Yes, thank you so much. So B and O are the possible blood groups. How did we know that? Well, basically, you and I will both know this because um, we have a Punnett square in front of us. Wait, where's my mouse pad? There we go. So it's either BO here, okay, BO here, all right, and on this side it's OO. Therefore, since both of them are recessive traits, they're gonna be there. If the child would have that chance of having, oops, and then another one here, OO. Okay, so let's assume my handwriting in that particular that particular illustration is fine. Okay, and how would you say so? Because Rins said 50%, how did she know that? Well, basically, one square is 25%. Okay, you're gonna be asked in the exams sometimes. I don't know if you're familiar with question banks, but in question banks, you're going to be asked, what is the percentage of the following? What's the percentage of the offspring of the following genotypes? If they're of the following genotypes. So they're going to ask, what are the blood groups, or what are the possible blood groups? What are the percentages of this? Blah blah blah. They're going to ask that, and each square is 25%. All right. So just multiply that to 4 and you're gonna get 100. All right, so if they're going to ask you these questions, at least you have the idea of the Mendelian principles. All right, so or this is also known as what you call the Mendelian principles because it relies on the allelic attribution of the um, of the traits. So we're gonna go and dissect those the terminologies that I've said one by one. Hopefully, you guys still remember your cytogenetics, which is I think third years. This is going to be taught in the third years. Am I correct? Ella Villamayor, was that taught to you in the third year? I really? Okay. Okay, so it was taken on her fourth year, so yeah. But when I was still studying, I think that was way back. I think that was way back 2007 when I first. I don't know when did we graduate, Miss Matulak. When did we graduate? I think that was a decade ago. I think that was a decade ago, but yeah. Yeah. So cytogenetics talks about these things. Unfortunately, it wasn't as detailed as what we're going to talk about right now. And this is actually one semester's worth of genetics review. So hopefully you guys can follow on with this because remembering the basic principles is important for you guys, primarily because these are questions that might be asked not just in your blood bank, but also in other exams. Okay? I think analysis of urine and body fluids will ask you questions about haploid and heterozygous, something related with genetics as well, if I'm not mistaken. So hopefully you guys you can absorb the information that I'm going to relay to you later today. So how many chromosomes is present in the nucleated cells of humans? Anybody can answer me? How many chromosomes are present in the human nucleated cells. Now I know I've already sent you guys a copy of it. I've sent you a copy of this particular lecture, but try to challenge yourself. How many? 46. All right. And how many of those is a yes, she's correct. Angela Felipe answered correctly. Thank you so much. That is 46. Now, how many of those are autosomal? You can answer pairs. I don't mind. You can answer in pairs as well. 46. So how many of those are autosomal? Autosomal chromosomes. Anybody? 22 pairs. Ina Joseph answered correctly. And how many pairs are sex chromosomes? Wonderful, wonderful. All right, so the answer to the question is 22 autosomal pairs and one pair of sex chromosomes. So you guys are correct. Okay, very good, wonderful. Friendiza on Tog. Yes, I don't know if I'm saying your name correctly, but yeah. Okay, so 22 autosomal pairs and one pair to relay the information for your sex. Okay, now let's move on to the next one. This is the polynucleotide molecule that carries the genetic material of an organism. Ooh, this one is a little bit different because a lot of books define a lot of books present the question for this particular slide differently. But yeah, let's try to dissect the questions one by one. Polynucleotide. Nucleotides, we have four types of nucleotides if we're talking about biochemistry. But I remember there's five. So what's the answer for this question? I'm gonna give you guys the clue. Nucleotides includes uracil, adenine, thymine, all right? What else? Adenine, thymine, guanine, all right? Cytosol. Okay, so what are those molecules? What do you call those molecules when they're bound or stringed together? That would be the answer to your this particular slide. Uracil, okay? Sorry, that's for a different one. Let's remove uracil. Adenine, guanine, um, cytosol, and Adenine, guanine, thymine. What is that? What when they're stringed together in a when they're stringed together or connected together in one line or one sequence, what do you call them? Because the question says polynucleotide molecule. Polynucleotide molecule. You have four types of nucleotide, and if they're stringed together in a sequence or a line, they would be called as such. Anybody can answer this question? Try to resist the urge to find the answer. But if it's okay with you, but if it's okay with you guys, you can search Google if you want to. Anybody? Let's try calling people from the chat. There's 50 people here. Wow. All right, let's try with the first person in my list. Almira Mendoza. Almera Mendoza. Hello. Do you know the answer to this?
Hindi po. Hello?
Yeah,
Hello
hello.
po?
Hello. Yeah, I can hear you. All right. Thank you so much for participating. And thank you for asking your chickens to answer for us. They're saying correct. All right. So the correct answer is DNA. Now let's define the following: gene and locus. A gene is basically what I'm talking to you guys about. It's basically a gene is a sequence in the chromosomes. Okay. If you're not familiar with the chromosomes, those are the X-looking figures that you definitely see in your genetics books or any biology related books, those X figures that looks like forks. All right. So genes are basically the a sequence in those things, a segment of DNA strand. Okay. Now a locus is the location of a particular gene in your chromosome. So for example, the H gene is located in chromosome 9. Sorry, 19. Okay, the AB gene or the ABO gene is located in chromosome 9. So the locus is the specific chromosome that we're talking about. All right, it could be either on the short arm or the long arm. It doesn't matter. All right, as long as you guys know the definition, a gene is a segment of a DNA strand that codes for a specific trait. When we talk about trait, this is something that will be expressed. Okay, when it's expressed, it is detectable. All right, we will talk about traits later on today, especially when we're doing blood typing of a patient. Now, let's move on to the next slide. These are genes located in a specific locus that differ in nucleotide sequences in individuals of the same or related species. So, for example, Almira, Zyrene, and Hannah can have one specific gene that differ from one another. Or let's go back with the discussion of Justin and Hannah. So this is, I'm gonna pop up the paint again. Hopefully you guys can see it. So these are the traits that we are talking about. It's B or O. All right, okay, in this case, okay. So when you combine these two things together, they're located at a specific location in their chromosomes. Okay, so they have a gene for B or O, but O is amorph, but we will talk about what is an amorph later on. Okay, so they're located and they can be changed. So Almira can have an A gene and can express H antigen AH can express A antigens. All right, and Ella can have AB. So because she has different types, and this is known as your alleles. All right, allele. The alleles in this particular combination is your B and O or BB or OO. That is an allele because these things can vary on different people. So I'm type O. I could have I can have an OO allele, which is homozygous. Okay, and then Justin can have the following: BB or OBO. All right, BB is the homozygous form, while BO is the heterozygous allele. All right? I got disconnected. Sorry. Sorry, I got disconnected. My microphone got disconnected. But yeah, do you understand the language? Yes or no? Do I need to explain it to you guys further? Oh, oh, I think I got disconnected. Nobody's answering. Hello? Did I get disconnected? Oh, but you guys get the idea. Do you guys get the idea of what an allele is, what the gene is, what the locus is? You guys are familiar with these terminologies. The next thing that we're going to talk about are the two types of alleles. All right, so the homozygous allele and the heterozygous allele. So by owing to our first discussion late earlier, we have what? Homozygous allele. Okay, homo coming from the word from the Latin from the Greek or Latin, I forgot which one, but it basically means it basically means same. Hetero means different. So please don't forget the suffixes and the prefixes because they might help you out when you're trying to think of what's the answer. All right, so homozygous allele from my previous example, BB is a homozygous allele, while BO is a heterozygous allele. Why? Same, same BO. Sorry, different. So BO, all right, and then BB are same alleles. Get it? Clear? Can we move on? Can we move on? All right, wonderful, Hannah. All right. This is going to be important primarily when you're going to ask a question about paternity on the blood. Anyway, okay, hello? Okay, yeah. So I ended something. I disconnected everything except for my mouse. All right. Okay. So yeah, I think we're fine now. Okay. All right. All right. So the number of copies of a specific gene that is present in an organism's genome is basically what? Is basically the let me check if my is my microphone okay now? Can you guys hear me correctly? Hope hello. Okay. So I disconnected everything that's connected on the USB hub except for my microphone. Hopefully my my voice can be heard in the discussions. So now, before we were so rudely interrupted, we were talking about this particular slide. This is the number of copies a specific gene is present in an organism's genome. The term for that one is gene dosage. We're going to talk about that later when we discuss the MNSS blood group systems. All right, because these are the ones that will dictate how much how much of a particular antigen one person can get. Dosage can actually be important. This dosage of a gene is actually important for you guys as well. All right. So now, what are the applications of gene dosage in to gene expression? So let's try to go at them one by one. Gene products, they are directly proportional to the amount of gene products that are produced by cells. In this case, these are the RBC antigens. Gene dosage can also result in phenotypic consequences such as deletions and insertions. All right. When I say deletion, let's go back to our discussion earlier. A deletion in a gene segment means a removal of a particular segment in the gene. An insertion is a removal of a particular, or an insertion is basically an addition of a particular segment of a gene during a mutation. All right, so basically we're talking about mutations here. All right, so changes in a dosage can result to a consequence in the phenotyping of a patient. We're gonna go deeper into that when we discuss the different blood group systems later on today. All right, hopefully we get to discuss all of them because there's a lot of slides actually. Okay, so now let's talk about the total genetic composition of an individual. When we talk about the total genetic composition of an individual, the term that we need to know is the genotype. All right. So I will ask a question from the class based on our initial discussion earlier. Based on our discussion earlier here, I'm gonna show the Punnett square again. Okay. Which one is a genotype there? Can anybody tell me the genotype? Is all right. So anybody can answer me? Which one is somebody answered? Angela Felipe answered, sir, yung nasa loob po ng squares. Um, yeah, you're correct. That's the genotype. Now again, what are the single ones outside the squares? These ones over here. What are these? These are alleles or traits, right? So the genotype of that person is this one. Now, can we say the blood type of the patient on can we see the blood type of the patient in this case? And Angela, Angela, are you there? Can we make assumptions? Yes. Why? Because these genes are already yes, the probability. Yes, correct. We can have an assumption of the patient's gene of the patient's phenotype or blood type based on this particular slide on this particular illustration. Why? Because the next thing that we're going to talk about is the next thing that we're going to talk about is the detectable or the expressed genes of the individual, which is your phenotype. Or in blood bank, this term is synonymous to the patient's or the person's blood type. All right? That's the reason why we have blood typing. Okay? Ay, dumating na nagulat ako, sorry. Naloka ako. Dumating na kasi ang sahod. Charing. Char. Kaya ako nagulat. Anyway, ang saya-saya. All right, so let's try to complete a particular card. you will see a lot of these things. Okay, I I will go around the class and ask people. Ay, nawala. Hello? Hello? Nawala. Ay, sorry. So I'll go around the class and ask people, is the presentation a genotype or a phenotype? Anybody can answer. Unmute yourself. What is the answer for this one? Complete the card. Alleles. Allele. We're talking about allele. AO and AA. Is it a genotype or a phenotype? All right, thank you, Ina. Okay. Before we answer, we'll try. We have to have a house rule. I forgot to tell you guys. You have to raise your hand, and then when I call your name, that's the time we can answer. All right. Anybody agree? So we don't have a lot of people answering at the same time. Clear? Will that be clear for everybody? Okay. So if the patient's blood type is type O, that means that the patient's blood type is termed as what? The genotype or the phenotype? What's the answer for this one? Is it the genotype of a patient's phenotype? Anybody? All right, let's try calling people from the class. If nobody's answer, I'm gonna call people from the class one by one. Let's try with somebody's raising. Somebody unmuted themselves. Or no? I thought. Anybody? Any takers? What's the answer for the second one? Is it the genotype or the phenotype of a patient? All right, Angela, give it a go. Okay, so basically some of the members of the chat know the answer to the question, which means they've understood the concepts of genotype, genotyping, and phenotyping. All right, let's go on to those are the answers. Okay, now let's. So basically, when we talk about genotypes, we're talking about the patient's inheritance. All right. So because these things are genetic, these things are related to concepts about genetics. We're talking about the inheritance, and your genotype is actually from your mother and your father, your biological. Not just I'm sorry if I sounded a little insensitive for some of the people here. Your biological mother and father. Okay. So biology can dictate basically what your blood type is, primarily because it is an inherited trait. Okay, so your blood type is inherited from both maternal and paternal genes, and your genes come out of from your one from come from one part of your genes come from your mother and another one from your father. Hence the term haploid, which means half. Half, guys. Please don't forget that one. Okay. Humina po tapos choppy. Okay. All right. Okay na ngayon. Let me change my hang on a sec. Let me change my headset. All right, so is everything clear now? Hello? Hopefully it's clear now. Let me modify the settings for the speakers. Oh, all right. Hello. Ayan. Ayan, okay na. Okay na, guys. Mahina, malakas.
Ayan, sir. Malakas na po.
Nag lumipat ako sa Chipanga na ano eh. Lumipat ako sa Chipanga. So please bear with the I will try to raise my voice. Is my voice unnervingly loud? Is it okay? But is it okay now? Hello. Ayan. Ayan, okay. Is my voice unnervingly loud? Hopefully hindi sobrang lakas ha. Ayan. Okay na, Angela? Okay, wonderful, wonderful. Si Angela talaga yung tinanong ko, no? Alright. Okay, so let's move on to our next topic, which is basically the two types of chromosomes. We've already discussed this. I think this was a repeat question. So we have somatic chromosomes and sex chromosomes. So we have 22 pairs, and then one pair for sex chromosomes. Now, let's go on with the difference between a dominant and a recessive gene. So basically, a dominant gene is a gene that is phenotypically observed as a trait, whilst a recessive gene is a gene that cannot be observed, but nonetheless, the person or the organism has this particular gene present in their genome. All right. So what do I mean by this? Let's
Sir,
go.
excuse me, di po. Ang hina, sir. Parang walang naririnig.
Parang walang naririnig. Talaga, Hana.
Nagcha-choppy po. Parang nagcha-choppy po, sir, yung voice nyo.
So lumipat kasi tayo sa Chipanga. Alright, testing, testing. Okay na.
Yan po, sir. Okay.
Dapat pala dito ako sa butas nagkasalita. Alright. So, please bear with the Chipanga microphone. Alright, so where were we? Dominant traits are genes that are physically observed as a trait.
Sir, choppy po.
Ayan. Mm-hmm. All right. Okay. Ayan na. Okay na, guys.
Sir.
Okay na po, sir.
No, I don't think it's okay. I don't think it's okay because I. Hello.
Mahina po, sir.
Here when I put it here. All right. Hello. 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 Okay. Testing. One, two, three. Microphone is okay. Okay, so you guys can hear my voice now?
Yes po.
Ayun. Okay, so yun lang pala kailangan kong gawin. Anyway, nasa na tayo? Difference between dominant and recessive gene. So dominant is a gene that is phenotypically observed as a trait, while the recessive gene is unobservable. However, it is nonetheless present in the organism's genetic material or genome. Okay, so what do I mean by this? Let's go back to our earlier discussion of patient type O and patient type B. Now, let me pull this out, these two together, and then I'm gonna enlarge this one. Now, which one is the the dominant trait, do you think? So we're talking about alleles here. We have O and B alleles. Let's focus on the B one first. Let's focus on type B. Which one do you think is the dominant trait here? Anybody can help me out? Okay, so Earl Palmero.
Sir, wala po yung presentation, sir.
Ay, baliw. Oo nga naman. Sorry naman, Earl. All right, so let's focus on our discussion earlier. So I enlarged the picture. All right, so you guys can see it. Can you guys see it now? All right.
Ano po?
Okay, so Earl, since you willingly lent your voice to the class, which one do you think is the dominant trait here? B or O?
B po, sir.
Why do you think so? By the definition.
B, sa, mas, mas madami po yung B, gene.
Mm, it's not what I'm looking. That's not the answer that I'm looking for. But thank you for participating. And your first answer was correct. You're correct with the dominant trait being B. Let's find another person who can help us out. What about another person in the class? John Mediana, please help us out here.
Homozygous, sir.
Huh?
Homozygous B.
Homozygous? Homozygous B. No, we're talking about genetic traits that are dominant and recessive. Let's go back to the definition of let's go back to the definition of dominant because I think a lot of people are often confused with what is the dominant and what is a recessive gene. Oops. Okay. So what is the dominant gene? What is the recessive gene? Um, let's go back to our discussion earlier. This one, okay, to be more specific. Mhm. So how do I know B is dominant? Well, this one we can't know, we can't tell. The first one. This one we can't tell because this is BB, right? This is a homozygous trait. Okay? But but may malaking but, guys. This one, on the other hand, has BO. It's heterozygous. We can know which one is the dominant trait here. And Earl answered earlier that B is the dominant trait. How do we know this? Because B, even in the presence of the O phenotype will not be represented. All right? It cannot be typed. Why? Kailan ba ako nag-blood type ng isang patient na, for example, BO na ang result niya is BO? Hindi, 'di ba? Wala tayong ganun, 'di ba? Kasi nakatago lagi siya. This is the reason why the recessive gene is also termed as the carrier gene, or sometimes the invisible gene, because it is removed from the equation once you're typing it in the blood bank. Do you understand now, guys? You can understand what's the difference between the dominant and the recessive gene? Or do I need to elaborate further? You understood na, Angela?
Elaborate pa po, sir. Para
Okay,
siguro
so let's try
kabalo.
again. Let's try again. What about if I say I develop? Okay, so that's a good question. That's a good answer. That's a good answer actually. Let's try and erase most of the things that I've done. Let's go back again. Say for example I have O this time. We're going to talk about O this time. Okay? O gene, as I've said earlier, is a recessive gene. How do I know this? Because in this one, O is not represented. But when it became codominantly represented as O, that's the time it became presented. That's the time you will be able to type a patient as type O. Nagets nyo? Nakatago lang siya kapag nandun ang presence ng B, ng A or B, sorry, kapag nandun ang presence ng A gene at saka ng B gene. Gets? In the ABO blood group system, the A and the B gene is much more dominant than the O gene because it cannot be typed or presented. Gets? Do we want another
Ba-
example?
Bali po, sir, kapag ka may kasama yung O, recessive po yung O. Kunwari po
Yes.
yung mga A sa B gene. Pero kapag ka OO lang, dun lang po magiging dominant yung O.
Yes. That is correct. That is very much correct. Wonderful analogy. Gets? All right, so I'll give you guys an example of a great example of this one. Imagine nagsuot ka ng maskara. All right? Um, Angela ka pa rin, 'di ba? Tama? Hindi naman nagbago, nagsuot ka pa rin ng maskara, Angela, 'di ba? Nagsuot ka ng
Opo.
maskara. Halloween, no? 'Di ba Halloween
Opo.
ngayon? Halloween ang usong holiday ngayon, magki-Christmas na. Okay, so nagsuot ka ng maskara. Angela ka pa rin, 'di ba? Gets mo?
Opo.
Pero nakasuot ka lang ng maskara. Ano yung nakapresent sa harap mo? Yung maskara mo, 'di ba? Yun yung nakikita ng mga tao.
Opo.
Gets mo? All right, so in that analogy, ang maskara is type B, is the B gene. Pero nakatago pa rin sa loob yung pagiging Angela mo. Nagets mo?
Yes
Do
po.
you get the analogy? O, di ba tinagalog ko na kasi medyo nahirapan kayo, guys? Nagets mo, John Mediana, kung ano pinagkaiba?
Yes po, sir.
Alright, can you please explain to me again?
Pag, pag opo, sir, parang hindi po siya
No,
na-express.
okay. Kapag, hindi siya na-express kapag,
May kasama pong B tsaka A, ganun.
Ayan, wonderful. All right, let's go again to Anna. Do you understand what we just said, what we just discussed, Anna?
Yes po.
All right, can you explain what's the difference between a dominant and a recessive gene?
Katulad po ng sinabi nila kanina,
Oh,
kapag si O itself, pag Oo, saka lang po siya ma-express pag may kasamang iba, like A
all
or
right.
B.
All
Mas
right.
ma-express po yung iba kaysa po sa
Okay. So pag nagsuot ka Ena ng maskara, si Ena ka pa rin, 'di ba?
Yes.
Regardless kung anong gawin ko, kung anong maskara ang suotin mo, si Ena ka pa rin. Ang pinagkaiba lang kasi, mas dominant kasi, mas nasa harap kasi yung maskara. Kaya yun yung nakikita ni April, yun yung nakikita ni Elia Villamayor, yun yung nakikita ni Earl Palmero. Alright?
Yes po, sir.
Okay, so wala tayong hugot doon ha. Hindi ako humuhugot, guys. Ay, ano ba yung kinlik ko? Sorry. Okay, so wala tayong hugot doon, guys ha. Alright, so can you guys see the presentation? I clicked on the presentation. Can you guys see it? Or is it still paint?
Nasa paint pa din po, sir.
Alright, so lipat tayo sa presentation. Alright, okay. So next, this is actually one of the most confusing things to students during my brief tenure teaching genetics before. A lot of students have problems understanding the concepts of recessive and dominant genes, and how much more would it be more difficult? I think it would be much more difficult if we go to the different blood group systems if you didn't understand it. That's why I took some time to discuss this specific topic to you guys. All right, now let's go to the expression of two dominant genes on the same allele. Oh, diyan na papasok si AB blood group system. Now why is AB blood group system considered as a codominant gene? Codominant gene. Let's go back to paint. All right, let's go back to paint and I'll show you guys the difference. Stop presentation, and then we're gonna go with another paint again. New window, paint. Now, when we're talking about codominance, let's go again with the new one. Don't save. Let's go with AB. Okay, AB, A, 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 O. Now, you might get a question like this in your board exam. You might get a question like this in your board exam. Don't be confused. It will ask you a question, which of the following, which of the following are dominant genes or codominant genes or expresses a codominant gene? Now, it will ask you a question, which of the following expresses a codominant gene? Okay, so that is the question. Let me enlarge it so that people can see the question. Now, I want somebody in the class to answer me again. Which of the four genes that you can see here, we have A, we have B, we have O, which of the three genes rather, which of the three genes that we have here are dominant? Anybody in the class? I've mentioned it a couple of seconds ago. Um, let's try asking a student. Let's go with the last one that I asked. The first one that I asked earlier. Angela, give me the genes there that I presented in the. Angela, are you there?
Yes po.
All right. So give me a gene, just one, among the alleles that I presented to you, give me just one that is a dominant gene.
AB po,
A lang. A lang. Isa lang naman ang tinatanong
AB
ko
po.
eh.
Ay, sorry,
Isa
sorry.
lang ang tinatanong ko. Masyado ka advance. Isa lang. A lang muna. Okay, so we'll start with A. What about John Mediana? Give me another one that is a dominant gene there.
B.
B, di ba? So we said A and B are dominant genes. Correct? Correct, John? Ay, natahimik si John. Okay.
Yes, sir. Yes, sir.
Sabi niya, sabi siguro ni John, ayoko na sir. Naloloka na ako sa iyo. Okay. All right. So, when we talk about dominant genes, A and B are dominant genes. I've mentioned it before, right? We're going to enlarge these two so you guys can see it much better. Oops. All right. So, we're going to enlarge these two and I'm going to separate A from B. Now, these two genes are dominant by themselves, all right? But when you put them together, they will be considered as codominant. All right? So this is a codominant expression. Why? Because there's two dominant genes that are still there, that are there in the alleles of the patient. What about in this one? Is this a codominant expression? This one, I'll put it here on the upper left side. Let's ask another person in the chat. Um, what about Earl? Earl Palmero.
Yes, sir.
Is true or
No po.
false? Is AA a codominant expression? Hindi pa ako nagtanong,
No,
sumagot
sir.
kagad ng no.
False.
No,
False, sir.
but Earl, I just mentioned earlier that A and B gene are considered to be dominant, 'di ba?
Domin... Uh, okay, sir.
Uh-huh. So, would you change your answer now that I've just said earlier?
Yes, sir.
Okay. So, Ella Villamayor, will you agree now that in Earl's changed statement that AA is now a codominant expression? Ella. Ay, wala. Hindi nagsalita si Ella. Let's go with somebody else in the class. Let's go at the very bottom. Cyrene. Cyrene, are you there? Ay, wala. Walang tao. All right. Who's here? Raise your hand. Wala gusto magsagot. Kaloka kayo ah. All right. So would you who would say okay, so para hindi na lang tayo nag-iisa-isa na students. Type on the chat if you agree. Is this is a codominant expression or no? Anybody in the chat? Anybody can chat. Type. Yes, sir. You can say yes, sir. You can say no. Yes, if I agree that it's codominant expression. No, if I don't agree that it's a codominant expression. You can type it in the chat. I will give you guys a minute to think about it. All right, whilst I drink a glass of water. Okay. Okay. So you guys have 1 minute to answer my question, huh? Yes, that it's codominant. No, if it's not codominant. All right. I will come back and hopefully people will be participating. Okay, let's see who answered. A lot of people said no. Only Angela Felipe said yes. Angela, parang feeling ko talo ka dito. Angela, anong masasabi mo? Lahat sila nag-disagree sa'yo.
Baka po majority wins.
Actually, Angela, you are the only one who said the answer correctly. I'll
Hindi
give
po
you,
pala tama yung majority.
yeah, not all the time majority wins. As I mentioned before, A and B genes are dominant traits. Okay? A and B are dominant traits. Okay? A and B are dominant traits. Now, when both A in the homozygous form, regardless of what form it is, it will become codominant. But what do I mean by codominant? I'll give you guys another analogy. Um, are you guys playing volleyball? You guys play volleyball, right? Who plays volleyball? Who plays volleyball here? Renalyn Amparo? Yes, thank you for participating. Renalyn, can you unmute yourself? Let's talk.
Yes po, sir.
Let's talk. Let's talk with each other one by one. All right. You play volleyball. Now there's always going to be a team captain, right?
Yes po.
But in the absence of a team captain, you will have a co-captain, correct?
Yes.
Yes. Ngayon, kapag magkasama silang dalawa, you will call them co-captains, correct?
Um,
'Di ba? Co-captain sila because they
okay,
exert the
opo.
same type of leadership. They exert the same leadership roles to the team. Is that correct?
Yes po,
Oh,
sir.
so since leader sila, sila lang ang nakikita. Tama? Sila ang nagre-represent sa buong team. Tama? Correct?
Sometimes po, yes, sir.
Sometimes po yes, sir. Sometimes po yes, sir. Pero in your experience, kapag ka sinabing si team captain at saka si co-captain magkasama, nakikita sila lagi sa mga pictorial, 'di ba? Even in
Opo, sir. Mas, ano
matches.
sila,
Most of the time they will represent
mas na,
the right?
represent.
Right? So I used
Opo,
volleyball
sir.
because in basketball I don't see a lot of co-captains in basketball. I don't know why. But yeah, that's the analogy I'm using here. So captains will be considered as somebody who will be seen all the time, and therefore they represent in this analogy, in this particular analogy, they are the ones that are often seen when they are blood type. Correct? Gets nyo? So pag pinagsama ang co-captain at saka ang captain, sa pictorial, sila lagi ang magkasama, nakikita sila lagi. And therefore, co-dominant silang dalawa, which means nakikita sila lagi. Gets? Nagets na? Nagets na ninyo yung analogy na ginamit ko? Magkasama lagi sila, co-captain, therefore, lagi sila nakikita sa pictorial. And again, in terms of blood banking, they are expressed at the same time. So kaya yung tinawag na codominant expression. Nagets nyo? So in A and B, because A and B genes are dominant, when we type it in the blood bank, that's the time you will see the expression together. Hence, codominance. Gets? Guess, dinodominate ang phenotyping. Now, we're not going to talk about which one is much more powerful, which one has a stronger reaction, because that's a different discussion again by itself. All right, now we're gonna move on to our next topic, which is yeah. So the example of a codominant expression is AB. We're gonna move on with the blood group systems. Okay, so location of rather just the location of the blood group systems. So basically, the questions for this one are quite tedious. It's more of memory work rather than analysis and analogy. So hopefully, many of you are good at memory games because in this particular set of slides, you will see a lot of which of the following is located, which of the following belongs to what particular chromosomes. So in our first slide, the major blood group system that is located in chromosome 1. I gave you guys a mnemonic, Ruffy, because it's the RH blood group and the Duffy blood group systems. All right? So we're just gonna go skate by this. It's quite tedious if we want to memorize it one by one. And then the major blood group system in chromosome 4 is the MNSS. How do we remember it easily? There in chromosome 4 in MNSS blood group system there are uh, four genes that we're looking for. The M, N, and SS genes. The big S and the small s. So there's four. Chromosome 4. All right. Blood group systems that are found in chromosome 7. Kel. I don't have a mnemonic for that one, unfortunately. All right. Which blood group system is found in chromosome 9? As I mentioned earlier before, it's the ABO blood group system, the ABO genes. All right. So the ABO genes is actually found in the in the in the chromosome 9. But sir, akala ko sir, amorph kanina sinabi mo ang O. There's an O gene, but it is not usually expressed. We'll talk about that later when we go in the specifics of the ABO blood group system. All right, so major blood group systems that are often found in chromosomes that are seen rather not that often found that are seen in chromosome 18, the kid blood group system. Mnemonic for you guys is that when you're 18 years old, you're no longer a kid. O, di ba? Pinapadali ko ang buhay ninyo. Hindi ka na bata kapag ka 18 years old ka na, isa ka ng adult. You are capable of making your own decisions. O, di ba? Ang ganda-ganda ng ano, ang ganda-ganda ng wala na tayong presentation Charing. Hindi ko lang nalipat. Sorry naman Sorry naman. Ayan. Alright, so mnemonic natin, you're no longer a kid when you're 18. You are You are capable of making your own decisions now. Alright? So that is my own mnemonics for you guys so you can remember. Is this important? Do they ask this in board exam, sir? Yes, they do. If you check the question banks for both ASCP and for the local board exams, they will ask questions about this. Okay? Now let's move on to the 19, chromosome 19, H gene. All right? H gene, sir. Akala ko ba H gene, sir? Tapos kanina may O gene pa, sir. You will know later on what's the function of the H gene. But just to be clear, there's also two other blood group systems that are associated with the chromosome 19, which is also the Lewis and the Lutheran blood group system. Now, the major blood group system that is found in type 2 is P1PK. We'll talk about them in detail later on. And then this is the summary of everything that I've said. Please remember them by heart, hopefully, is specifically chromosome 9 and chromosome 19, because this one is the important ones. Chromosome 9 and chromosome 19. You have to remember these ones. And also in chromosome 1, which is basically the RH blood group system. If you were to ask me, I think the most important thing that you need to remember is the one for chromosome 1, the one that's involved for the RH and the ABO blood group systems. Okay? Many a times I've encountered these questions in the exams and in many practice exams, and hopefully you will not make the same mistake as I did, because when I took the board exam I didn't care about these things, and there was one point that I missed because of this. All right? Because I remember the question is which of the following chromosomes contain the genetic material for the ABO genes. O, di ba? Sayang yung 1 point. So, kaya every time na may discussion sa blood bank, laging pinag-uusapan yung chromosome numbers na yan. So, yun lang. Okay? Yun yung summary ng lahat ng mga pinagsasabi ko. Now, why did I put it at the back? Because I want you guys, when you go home, when you go home, you're already home. I forgot that this lecture is being conducted in work in your homes. When you're in your own time and you're studying for the exams, I want you guys to challenge yourself. Stop looking at the second slide, the answer slides, but rather check in with try to challenge yourself in remembering these things, because I know this can be very helpful, especially when you're developing when you're developing your analytical and your memory skills with with a lot of things. I think this is one of the reasons why a lot of my friends used my flashcards when I was in college, because they really don't have the time to memorize everything, and the fastest way to do it is by memorizing by via flashcards. And that's how I did, how I passed the exams. Hopefully, some of you might use this in the future. But anyway, let's go on to another review. This is now, we're going to go with the review of immunology, because a lot of the concepts in immunology is important in blood banking. That's the reason why it's the name for the official name, or the scientific course name for blood banking is not actually blood banking, but rather it's immunohematology, which means concepts of immunology combined with the knowledge of the blood. All right. So let's review immunology for a while, with with a certain topic, with select topics that are usually discussed in the blood bank. This is just a review. What is the most important concept that is applied in blood banking? Immunology concept that is applied in blood banking. Can anybody answer that? Anybody in the chat? What discussions ba ang pinaka-important in immunology that is usually applied in blood banking? Anybody? Let's try with somebody from the 49, 42, kasi nandiyan pala si Miss Matulak. Um, let's try with the meeting host, Ella Villamayor. Ella, unmute yourself. Ay, tinarayan ako. Hindi ako pinansin. Ella. Hello, Ella. Good morning. Wala. Let's talk to Harold Bermudez. Harold, are you there? Are you participating in our class? Hello? Hello? Nobody. Okay, let's try again with John Ilagan. John Ilagan, are you in the class right now? So there's three people. There's three people that I've
Yes
called.
po, sir.
Ay, hi, John Ilagan. Ikaw pala yan. Ikaw pa,
Good morning
hindi ko
po.
naalala yung ano mo. Good morning din, John Ilagan. Hi. Now, I have to ask you a question. John, you've studied immunology, correct?
Yes po, sir.
All right. So what do you think is the most important immunology concept that you've studied in your third year, I think? Yeah, I think in your third year, that is usually applied in the blood bank. That's most commonly applied in the blood bank.
Yung ano po, about sa immune system po.
Immune system? Really? Do we study the immune? All right. Thank you, John Ilagan. But I think that is the incorrect answer. But thank you for participating.
Thank you po, sir.
All right. Angela Felipe. You raised your hand, Angela.
Antigen antibody po.
Wonderful. Yes, that's correct. A lot of the discussion, yes, that's correct. A lot of the questions in a lot of the concepts that are, yeah, but John Ilagan is not entirely wrong, because without the immune system, or the without the immune system, we wouldn't have antibodies, correct? So he's a little bit wrong, but
Sorry, sir.
yeah. No, 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 no. I will try to give you at least a little bit of credit, but without the immune system, we won't have antibodies, and we won't have knowledge of how they're going to be formed. So, and so, as Angela and Cyrene have pointed out, the most important one that we usually use in the blood bank is the antigen and antibody principles, or the antigen antibody reactions. And because of the fact that it is useful in the study of blood groups, it is useful in cross matching and transfusion studies, and it is also it's also useful in resolving and explaining discrepant results. So we're going to talk about discrepancies later, but for advanced analysis, for advanced study, what do we mean by discrepant results? Anybody in the chat, anybody in the class can help me with what is a discrepant result? Let's go with ABO typing na lang para mas madali. Kasi this is the only one the only one where we go into detail when it comes to discrepancies. What do I mean by a discrepant result in the blood bank? There is an anomalous result and then there's a discrepant result. We'll go with the discrepant result first. Can anybody in the chat tell me what's a discrepant result? I think I should give also a monetary compensation to the person who will be able to answer my question. Last in the yesterday in my class in microbiology, the students aren't as active because there wasn't a reward. So I asked Sir Marco if I can give a reward to somebody who can answer the toughest questions in that I will ask. For um for I think 50 peso Gcash load, anybody here can describe, can even in Tagalog, you can say it in Tagalog if you like. What is a discrepant result? Anybody for 50 pesos? Oh, kita mo na sabi ko na kailangan ko lang magbigay ng ano eh. Kailangan ko lang magbigay ng monetary compensation. I forgot the term. Remember guys, malapit na birthday ko kaya mabait ako kapag ka birthday ko. Ako actually namimigay ng gifts kapag birthday ko. If hindi po match yung forward and reverse typing. Ayon! Correct, Zyrene! Correct. Correct, Zyrene. That is a discrepant result. That is correct, Zyrene. That's the one that I'm looking for. If the results aren't confirmed by if the forward results aren't confirmed by reverse typing. That is correct. Thank you so much, Zyrene. Now I want you disagreeing with each other. It's not very detailed. The most detailed answer was from Zyrene Alvarez. Unfortunately, from Friendiza. All right. Now, guys, I will also ask you. I will also ask the class to the participation in this particular type of topics. When I say I will give a monetary compensation, you will answer me like this. You will raise your hand like this. Okay. And then, you're going to say, Sir, namamas ko po. Para game show lang tayo, no? Sir, namamas ko po. And my answer is, blah, blah, blah. Ganon, no? Pero for Zaylene, I want you to email me your number later. Just like last time with John Ilagan, I asked him to email him my number, and then we sent him I think a thousand pesos in GCash. Although I don't know if John Ilagan has already received it. Let me confirm later on in after the discussion. But anyway, but
Thank you,
anyway,
sir.
but anyway, John Ilagan or John Mindiana, I don't know if it's John Mindiana. But I remember there was a
Diyan
John.
na po.
Midiana ba? Yeah, yon.
Yes po,
Opo,
sir.
sir.
Yeah, yeah. So hopefully John Mediana has received my 1000 peso prize from last week. From last time's lecture. Because next week there will be another contest which is worth 500 pesos. All right? So Zairine, email me your globe number. Okay, then you'll get 50 pesos. All right? Kailangan may monetary compensation talaga, no, para magano kayo, para mag-participate kayo, no. Nakaka-lurky kayo, ha? All right, now let's go to a true or false question. True or false? An immunogen is always considered as an antigen, but an antigen may not always be an immunogen. I want people to the chat. I want to see people typing in the chat. I will give a minute to the people. People will say true, 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 or false, false, false. Kung ano man nasagot ninyo. Alright, so Angela said true. Who else can participate? I'm timing. Now, 1 minute starts on 1 minute is in the clock. I'll drink water for a while because my throat is hurting. And all right, that's the end of our timer. Now let's see how many people said false. Because the correct answer is true. True, 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 true. Everybody said true. So majority of the students said true. Okay, wonderful. Wonderful. The correct answer is true. All right. Majority of the students answered true. Now, some antigens may not initiate an immune response, but they are considered an antigen. Okay. All immunogens, by the name itself and by definition, is an antigen that is capable of initiating an immune response. So yes. So the answer is true. Now let's move on to factors affecting the immunogenicity of an antigen. So we have six things to consider: foreignness, complexity, molecular weight, digestibility, dosage and route of administration, and accessibility of reactive sites. Let's go at it one by one. The concept of foreignness basically means this is your auto antibodies or auto antigens, allo antigens, xeno antigens. So I'm not gonna go and delve deeper into this because I know you guys have already discussed it in immunology. Okay, so just remember the terms auto antigens, antigens that are present in the host and can produce autoantibodies. All right. Allo antibodies and allo antigens are antigens that may vary from related species. All right. Xenoantigens or xenoantibodies are antibodies produced against another antigen, another species. For example, um, antigens that are derived from plant products or animal products. If we want to be particular, and then we have isotypes. These things are the ones that we need to talk about. These are things that are related to what we call foreignness. Now we move on to chemical complexity. Is it a protein? Is it a carbohydrate? Is it a lipid? Is it a nucleotide? That's what we mean by complexity. Molecular weight, anything that is greater than 10,000 daltons. This is something that would most probably initiate an immune response. Digestibility, if the antigen in question is capable of being phagocytized and being digested and then presented in antigen presenting cells, that's another thing that would determine whether or not the immune response that whether or not it is immunogenic. Dosage, when we say dosage, the amount of antigen that you've received. So for example, zero point 0.0.1 ml of 0.01 ml of mismatched cross mismatched ABO blood group can result to can result to an episode of immune of um, an immediate hemolytic transfusion reaction. So that is the term dosage. The route is either if it's either IV, which is the best site to initiate an immune response because that is rich with blood cells and immune cells, of course. And then lastly is the accessibility of reactive sites. Do we need to use enzymes to enhance the reaction? Do we need to cleave some parts of the antigen to be recognized by immune cells? So these are the things that these are the factors that can affect the immunogenicity of an antigen. All right? So clear? These are concepts from immunology. Hopefully, you still remember your immunology class because, as I've said before, there are some concepts, specifically the antigen and antibody reactions, that are important in the discussion of immunohematology or blood banking. Now let's go on with the complexity. Let's talk about the different types of antigens. So we have proteins, carbohydrates, glycoproteins, and glycolipids, lipids, and nucleic acids. They are arranged from the most immunogenic to the least immunogenic. So nucleic acids are actually the least immunogenic. So what again are nucleic acids? This is the guanine, adenine, thymine, and your cytosine. All right, when they are paired together, what's the term again? Anybody in the chat? What's the term again when you combine your nucleic acids together in a string to form a code? What is that? Anybody in the chat? A string of nucleic acids or a sequence of nucleic acids.
DNA.
DNA. Thank you so much, Earl. All right. So that is the correct term. Proteins are the most immunogenic. So please don't forget it. If it's not going to be asked in immunology, it's most definitely going to be asked in the blood bank. All right. Next. Let's move on to the next slide. How much should a substance weigh to be immunogenic? I've mentioned it before. It should be greater than 10,000 daltons. All right. The most effective route for an immunogen to elicit an immune response is anybody? Chat or the microphone. You can use chat or microphone. Answer this question for me. Anybody? I mentioned it a couple of seconds ago or a minute ago. The most effective route for an immunogen to elicit an immune response from a host is IV. Thank you so much. Thank you so much, Gina. Thank you so much, Angela. All right. Let's move on to let's move on to where are the active sites for blood group antigens located? So RBCs are primarily the locations of blood group antigens. Okay, but that doesn't mean it's just in the blood group. Okay, it's just in the RBCs. We have other blood groups that have even ABO blood groups. They can be expressed in the other cells as well. So even platelets, even lymphocytes, and even in epithelial cells. What is the main purpose of knowing the person's blood group antigen? Because if you know the patient's blood group antigen, you will know the patient's blood type or the individual's blood type. Okay? So knowing the patient's blood group or knowing the patient's blood group antigens or by typing the patient's blood group antigen either through forward or reverse, you will know the patient's blood group. Okay? All right. So now, it's a nice to know question. Based on a recent study, how many blood group systems have been discovered? There's actually been 36 blood group systems, and there are greater than 250 antigens that have been discovered. And up to today, there's still an ongoing study, especially during the COVID pandemic. There's actually a new I'm not familiar with I was supposed to put it here in our discussion, but I don't think it would be asked in the board exam. But during the COVID pandemic, they've discovered that there's an antigen that is in the surface of the RBCs, wherein wherein the COVID virus can actually latch onto. So see, so even up till today, there are RBC antigens or our blood groups that are being discovered because of molecular studies. Now, let's talk about groups of antigen proteins. What are they? We could have a single pass, we could have multi-pass, or we could have a GPI linked protein. Now I'm gonna discuss what is this um, a different in a different presentation. Wait, hang on a sec. Um, can you guys see paint? Not sure if you guys can see paint, but let me check. Ah, no, you can't see paint. Stop presenting. So we go to a window, and can you guys see the paint now? Anybody? Can you guys see paint?
Yes
Yes
po.
po,
All
sir.
right. So we have three terminologies to be familiar with: single pass, multi-pass, and GPI link. Single pass, multi-pass, and lastly is GPI linked. GPI linked. Okay? So these are the appearances or the biochemical conformation of protein protein antigens. Okay? Let's start with single pass. Single pass is like this. Imagine this circle. We're gonna have this circle. I'm gonna erase the bottom part of the circle. All right, so I'm gonna remove this one. All right, so that imagine that is the I want it pixelated, so I'm gonna go in. Okay, so imagine this is the surface of your cell. A single pass is basically something that is an antigen that goes down the lipid bilayer. That's a single pass antigen. All right, means it passes one time only. All right, and then when we go to multi-pass, let me select another portion of our cell. When we go to multi-pass, Shift C, Control V, need this one for GPI link. When we go to multi-pass, and then we're going to select a different color, it's an antigen that goes by one time, and then it loops back up again and again. That's a multi-pass. It means it passes several times, just like this. And then the antigenic determinants are located somewhere here, or somewhere here, and somewhere here. Okay? So that's a multi-pass antigen, right? And then for GPI link, there's basically a GPI molecule here. And then the antigen is, the antigenic determining site is either the whole thing or a portion of the GPI linked protein. Okay? So you guys you guys understand the difference between a single pass protein, a multi-pass protein, and a GPI like linked protein. Because each of them these terminologies will actually be used in the discussion in some of the protein-based blood groups. That's the reason why I added it here. So you guys don't get confused. What does it mean by single pass? Sir, what's that? Like seriously, mga ganon. Mga ganong mga questions. So we want to be familiarized with the language that we're talking about. Now, RBC antigens are detected through what test? The most simplest test. RBC antigens are detected through what process? Anybody in the chat? How can you detect the simplest way to detect RBC antigens? What is the simplest way to detect RBC antigens? Anybody? Okay? Nobody? Nobody will be able to answer? Earl, do you have any idea? Zyrene, do you have any idea? John Ilagan, are you still there?
Yes po, sir.
All right. So how do you detect RBC antigens? When you are a student, okay, si John Ilagan yung nag-react. John, John, where are you, John?
Yes po, sir.
John, paano niyo nalalaman sa class niyo dati kung paano mag-type ng blood groups?
Ano po, sa ano po, red blood cells po, sir.
Okay. Okay, so paano natin ginagawa 'yon? Can you discuss to me even in Tagalog, it doesn't matter. If you want to flex, you can say it in English, but in Tagalog, give me the procedure of how you do blood typing when you were a student. Give me a summary, a brief summary lang. Could be a
Ano
one
po,
sentence.
sir, parang yung blood sample po imimix po na merong may antibodies against type A and type B blood.
Yes, and that process is called what?
ABO typing, sir.
All right, that's forward typing. Yes, ABO typing. Okay. And the principle of the test is what?
Ano, sir? Pwede pa ulit po, sir? Ano
What's
po?
the principle of that test? What's the principle of that test?
Ano po, sir? Para,
Principle. I'm asking what type of what type of is it? Is it spectrophotometry? Is it what's the
ah,
other term?
sir,
Um,
ano po, agglutination reaction, sir?
what type of agglutination reaction? I like the fact that you're answering follow up question again, follow up question again, follow up question. What type of agglutination reaction?
Ano po, ABO?
No, no,
Ay,
no, no,
sorry.
no, no, no. There
Sir,
are
wait lang
several
po.
types of agglutination reactions, right? There are several types of agglutination reactions. You remember your immunology pala, hindi hematology.
Ano po, yung antigens po ay na-expose po siya sa antibodies, tapos nag interact
No,
po siya
no, no,
from
no, no,
visible
no. You're defining the principle. I'm asking the term.
ah,
Give up na, pagod na.
sorry, sorry sir, di ko po ma-recall.
It's okay, it's okay. The fact that you're participating is enough for me, actually.
Okay po sir.
Itaas
Thank
ang
you po.
bandera. Itaas ang bandera, no, John Ilagan. I love it.
Thank you po sir.
Oh, you're welcome. All right. Somebody in the chat already answered for you. Please thank them, John Ilagan, ha? Because Renalyn actually answered the question for you. Direct heme agglutination. So please thank him or her, her pala, sorry, her. Renalyn Amparo answered correctly. The correct answer is direct heme agglutination. Okay? What do I mean by direct heme agglutination? Basically, you're going to add anti-sera. The anti-sera has antibodies directed towards RBC antigens, and therefore you don't need to have a carrier protein. What do you mean by that one? It targets the RBCs. Heme agglutination. Heme ang tinatarget niya, the RBC, the red part of the sample, the red part, kaya nga heme agglutination. Why is it direct? Because you don't need a carrier protein. Indirect heme agglutination requires a carrier or a latex particle to agglutinate the substance. We have also another one, co-agglutination, and that is another terminology that is another principle by itself, another immunology principle by itself. But in this case, blood bank tests are usually done through heme agglutination. So please don't forget that, because that is one of the things that we will discuss. One of the things, or I think that's the only reaction, that's the only principle or blood banking or laboratory principle that you will encounter, except of course the other ones like illusion but I think that's the only thing that you will encounter throughout this whole discussion. All right. Wait, what happened to my slideshow? Ay, anong nangyari sa aking slideshow? All right, thank you so much for John Mediana. Nagaling-galing ni John Mediana. Every follow-up question, tinatry niya talagang i-answer. And I like that. And I like that when students do that. Why? Because it means that they are willing to answer regardless of whether or not they answered incorrectly. Because if you answered incorrectly, you will actually learn from it. Okay? In this particular lecture, I want you guys to feel comfortable and trying to answer the questions. That's why even if I know you're not going to answer correctly, I don't use that against you, all right? I don't use that against you. I want you guys to at least participate as much as you can, because this means that you're trying your best to answer the questions regardless of whether or not you answered incorrectly. Okay? So try your best to answer the questions, because this is for your benefit. It's not for my benefit. I've already taken the exam and I've already passed the exam. Aliagan ba yon? Sorry naman. Hindi pala ilagan. Liagan ba? Sorry naman. Okay, so thank you so much, John, for participating, ha? Okay, so I hope everybody would have the same tenacity uh, as your dear classmate John. All right. So now let's move on to why is it not presenting my I hate this. Anyway, let's move on with the methods of direct heme agglutination. Why is it not presenting? Grr. Ayan. So what are the methods of direct heme agglutination that we use in blood typing? So we have three methods, of course, that I put there. You can either use the slide method, the tube method, and the gel method. And actually, there's a grading system for each of these, but for the purpose of this lecture, since we're much more focused on answering board exam questions, I'm not gonna put the reactions, the plus one, plus four, plus five, plus one, plus four, plus up to zero. All right, so from 0 to 4, that's the strength of the reaction that you're going to get. Okay, so in the board exam, I've never really encountered a question that even in my practice tests, and I think your professors will agree with me in this. You will not get a question like that. You will get only questions like, this is the blood group of the patient. What is the problem, or what is the discrepancy? This is the antibody panel of the patient. What antibody is present? Something like that. Okay, so I don't think you will be asked questions like that. Now, let's talk about the major differences between slide direct heme agglutination and the other one. So we have gel method and tube method. What's the main difference with these with these three? Well, basically, slide is the only thing that's different. The results are reported as either positive or negative. And for tube and gel method, it's reported as positive or negative, but with the addition of a grading system. It's either +2 or +4. It could be a mixed field reaction. So that's much more somewhat semi-quantitative in a sense. So what does gel method look like? So I has anybody done a test in the blood bank before? I mean, has anybody actually participated in doing blood banking procedures like in the schools? Let's interview our your other classmates. Let's talk to Zairine. Zairine, hello, are you there? Let's go with a one-on-one. Now, are you
Yes po.
what type of blood typing procedures have you done in in the class when you were in college, when you were in your third years or fourth year? I'm not sure when they teach blood banking, but what type of methods have you done in the blood bank when you're typing for blood? Is it tube method? Is it slide?
Ulit sir.
What type of methods? At Tagalogin natin, anong methods ang ginamit ninyo nung third year kayo? Di ba? Tinagalog ko siya. Nahirapan na ako.
Shove and slide method po.
But you have not seen gel method, like the one that uses cards?
Hindi po.
Hindi pa. All right, so let me enlighten you guys what a gel method looks like. Let's go back to our hopefully ma-amaze kayo. But I don't think you will be amazed because I think it's pretty standard nowadays in many blood banks. Let's go back to our discussion. This is actually what a tube method looks like, or a gel method looks like. All right, so there's a gel column. Okay, can you guys see my mouse? In no, let's use a highlighter. There's a gel column, this one that I'm staining with with the yellow. All right, and then there's a cell button on the top. Okay, cell button is actually the reaction of the antigen and antibody reaction, which is basically going to give you either a negative or a positive. In this case, is this a positive reaction or a negative reaction? Anybody in the class? You can type it in the chat. I'll give you guys a minute to answer. Is this particular result a a positive one or a negative one? Is it positive or is it negative? That is the question. Positive, 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 positive. I'll give you guys a minute. I'll give you guys a minute, okay? I'll give you guys a minute as I drink my water. So yeah, we want to be as democratic in this class as much as possible. All right, timer's up. Let's see what the answers were. Majority of this class answered positive, positive, positive. Walang magko-contest, walang magpapakakaren. Walang magpapakakaren. Ayoko. In my opinion, based on my opinion, I'm entitled to my own opinion. I think it's negative. Walang ganon. Lahat magpa-positive. Okay, let's try to interview someone who answered. Rachel Tesoro. Hi Rachel, are you there?
Yes, sir.
All right. So paano mo nasabing positive siya?
Ah, kasi sir may agglutination,
Nasaan ang agglutination natin?
yung nasa ibabaw po, yung RBC.
All right. Paano mo masasabing negative ang reaction?
Pag wala pong agglutination,
Okay, so paano mo masasabi nga?
pag ano po, na-dissolve po yung RBC.
Na-dissolve ang RBC. All right. Thank you so much, Rachel. Glyza La Chica. All right. Unmute yourself. I want to talk to you. Good morning, Liza. Hi, how are you? You're on the line. For 500 pesos. Charing, for 500 pesos. Charing. Charing lang. All right, please explain your answer. Glyza. Uh-huh. And then, uh-huh. All right. So, so what do you say is the function of the gel? Glyza. But thank you, Glyza. All right, Cyrene. Bumabanat ka na naman, Cyrene. I would like to talk to you again, Cyrene. Cyrene.
Okay, sir.
Bumabanat ka, Cyrene. You meant to say strong agglutination reaction. All right, so what do you think is the function of the gel? Umalis na si Rachel.
Para
Natakot si Rachel.
mag-float siguro,
She left.
sir, yung RBC.
Anong ginagawa niya? Sige, Tagalog na lang tayo.
Para po siguro mag-float yung result ng RBC.
Nagfo-float. Nagfo-float. All right. Apparently, not a lot of people know what the function of the gel is. So I'm gonna help you. I'm gonna help you guys out. Is it okay for you to? Is it okay if I want to help you out? Okay, so Gina wants to help us out. All right, Gina. Gina, where are you?
Sir, yung gel po parang...
Wait lang, ang lakas. Nabingi
Hello
ako.
po, sir.
Wait lang, nabingi ako. Wait lang. Nakakaloka ka. Sobrang excited ni Gina. Binigi niya si Sir. Alright.
Sorry.
Ano yun, Gina?
Okay na po.
Okay na.
Yung gel po para siyang pansala. Yung sieve po ba, sir? Yung parang, yung malalaki lang po, yung strong agglutination, mapupunta sa taas. Tapos yung mga maliliit is yung sa baba po. or
That's
sa gitna.
good. Yes, that is the best answer. It's like a sieve. Ang arte. I love it, Gina. I didn't know there was somebody like you who existed in the class. Parang Siv. I love it. Yes, but that is correct. That's the correct answer, Gina. Thank you so much. So it's like a sieve. So there's like these are basically like microbead. There's microbeads inside, and if there's a any form of agglutination or strong lattice formation, that's the correct terminology. If there's a lattice formation, it means that the cells wouldn't be able to pass because of the small particles, because of the small holes within them. But if it allows single cells to pass through. However, if there's agglutination, as Gina have said, it would not be able to pass through. So again, that is like a sieve. Well, basically was what she meant. All right. So parang nagawa ka ng pansit canton, Gina. Is it like that, Gina? Gina, is it
Yes
like
po,
that?
sir, parang gano'n.
Parang gumawa ka ng pansit canton or parang gagawa ka ng ramen, yung gagawa ka ng udon, no? Itinadaan mo muna
Opo.
siya dun sa pangsala. Oh, thank you so much, Gina. Wonderful. I love it. Pero sayang, kung nasagot mo siya in the proper way, like, sir, namamas ko po. My answer is ganito. Ganun sana. Sayang, meron ka sanang 50 pesos. Sayang, Gina. Let's try again next time na, Gina. All right. So where were we? Before I was so rudely shouted upon by Gina. But no, char lang, Gina. Ha? Uh-huh. Okay. So what's the major difference between So there's grading methods. So what are the advantages of using gel cards? They are more objective. Okay, and you require only a small amount of sample. Um, in my in our protocol here in our hospital, we only need like 2.5 point 25 microliters of red cell suspension, and the stability of the cards can last longer. All right. So what do I mean by the stability of the cards can last longer? You can actually use it for investigation. So for example, nag-blood type si Gina, type niya type AB. And then si John Mediana, nag-type siya, naging type O ang pasyente. We can pull out Gina's results from 3 days ago. Remember, 3 days lang ang stability niya. Unless you put it in the refrigerator, then that will extend the life of the gel cards. And then we can compare, oh my gosh, nagkaroon ng clerical error. That's how you can use it for that's how you can use it for investigation purposes. So sometimes it's useful, and sometimes why do we need to do why do we need to allow the cards to stay longer in the laboratory? Because there are some cases, for example, in a lot of protocols, if you're going to follow the American Association of Blood Banking, they always want to retype the patient regardless of how many times the patient has been going in and out of the hospital. For example, manganganak na si Cyrene. Oh my gosh, jaundice na si Cyrene. Okay? Manganganak na si Cyrene. And unfortunately, the doctor noticed that her case is a CS case, or it requires cesarean section. So American Association of Blood Banking says that the Cyrene must give some sample in order for her to in order before she goes to surgery or before she goes to the operating theater. What do I mean by this? She needs to get pre-cross match sample before she goes to the so parang may reserve na. Gets nyo? So she needs to have a reserved cross-match blood just in case she undergoes some sort of just in case she undergoes some sort of complication during the process of the cesarean section. Because we may never know that once the OB gynecologist consultant slices into her, the consultant might have hit a large artery, and it might cause her to bleed out. So that's the reason why we need to have stable cards for comparison purposes. Okay, we're going to talk about that when we go to cross match investigation or mismatched investigation, or rather hemolytic transfusion reactions as well. So how we how we can how we can resolve problems like this, and how we can investigate something like this. Because in the blood bank, remember, as I posted before, everything that you do, as I posted in the previous in the previous slide, in the second slide to be quite specific, a lot of the things that you when the patient when the patient goes into either surgery or receives blood transfusions. Okay, clear, everybody? Okay, so actually, I'm gonna tell you a story. Um, it's I've been in the practice for almost 6 years now. I think 8 years, including my my brief tenure there in the Philippines, which is 2 years. I've only recently started working in the blood bank because uh, takot na takot ako. I'm scared to handle a patient's life. I would much rather go and work in microbiology because mas nauunang natetegi yung mga pasyente sa micro eh. Before you can release the results, the patient have already expired. And that for me is much more comforting than actually handling the patient and then uh, and then and then helping themselves save their lives in the process. And until recently, I think 6 months ago, I've started working in the blood bank. 'Di ba? 6 years na ako, almost 6 years na ako nagtatrabaho, pero ngayon lang ako nag-handle ng cases sa blood bank or ng samples rather sa blood bank. 'Di ba? Because it's the every test, I think every test meron akong kasama pag nag ano siya, nag-cross match siya, lagi siya nagdadasal. Kasi sana Lord wag daw sana magkaroon ng transfusion reaction. Yun yung lagi niya pinagdadasal. Alright? Lord sana, yun yung sinasabi, Lord sana sana yung in-cross match ko ngayon makatulong dun sa pasyente at saka hindi siya magkaroon ng any complications. So please be careful. Please don't take the blood bank procedures very lightly because although the this specific section among all the other sections in the laboratory, it pays the most, especially kapag nandun ka sa US. I have a friend there who works in the blood bank. Um, ang average entry level payment ng isang medtech is $21 per hour. But for blood bank technologists or technicians, it's around 35 to $40. So imagine the difference. So it's highly lucrative, but there is a bit of responsibility. So any mismatched needs to be investigated. Any discrepancies should be investigated. So yeah, next. How many days does the gel card remain stable? It's 3 days. All right. Okay. Now, arrange all the immunoglobulin classes based from the lowest to the highest concentration in the serum. So which one do you think is the most abundant in the serum? Just a general review. Anybody can answer. Which one is the most abundant? Is it IgG? Oh, somebody already raised their hand. All right. Um, Angela,
Sir, namamas ko po. My answer is IgG.
hindi siya kasama sa unfortunately hindi siya kasama because it's very easy to answer. Pero that is the correct format. Thank you. Thank you for showing your classmates the correct format on how to answer prized questions. But yeah, the answer is IgG. Now, in the immunology section of the exam, you will need to remember this particular mnemonic, GAMDE. All right. So this is basically the highest. Let's go here. This is from the highest to the lowest concentration. All right. Cool. Wonderful. 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 All right. No other questions? Anybody has questions? Anyone can contest what I'm saying. You can say, no, sir, I think what you're saying is wrong. In my opinion, nagpakakaren ka na naman. In my opinion, I would like to speak to your manager, sir, because I think what you're saying is incorrect. Ganon. No? No, we're talking about science here, so your opinions does not matter because your opinions are going to always going to be superseded by facts. So we're talking about science, so it's not an opinion-based class. All right. So now, immunoglobulin classes that are significantly or which immunoglobulin class are significantly encountered in the blood bank. Again, somebody from the class can answer. This is not a unfortunate. All right, Cyrene,
GMA po.
GMA. Sayang hindi niya sinabi. Akala ko sasabihin niya yung ano, sasabihin niya yung format natin. Pero pag mahirap yung question, pag binanggit ko na meron ditong prize, merong monetary ano to, compensation, doon kayo babanat ng ano ha, doon kayo babanat ng, Sir, namamas ko po, my name is ganto, and my answer is ganto. Alright? Next. Fragments
Noted
of
po.
fragments of the immunoglobulins. Alright, so we have two fragments. We have the FC portion and the FAB4 portion. All right, so what are the regions of the antibodies? We have the constant region, and then we also have the variable region. Is this important in the blood bank? Of course, yes. You'll see later why. Now, the immunoglobulins that causes hemolytic disease of newborns. All right? Ay, sayang, sana tinanong ko kung ano ibig sabihin ng HDN. Pero very common lang siya, madali lang siya masagot ng mga students. Kaya hindi ko na tinanong. HDN is hemolytic disease of newborns. All right? Which immunoglobulin class is the most? Oh, Angela, sige na, raise ng hand mo. It's very Hermione Granger of you, but okay. So which immunoglobulin class causes HDN? Namamas ko ka ulit?
IgG po.
Akala ko
Hindi
namamas
na
ko.
po. Hindi.
I-announce ko na pag pagka merong prize. All right? So IgG because it crosses the placenta. All right? It crosses the placenta. Now, wala dito sa notes natin. Ay, ayoko, feeling ko alam nila eh. Because wait, ire-ready ko yung clock para may challenge. Parang game show lang. Nakita niyo yung clock? Wala. May clock, guys.
Wala
Meron,
po.
walang clock na nakalagay. Sorry. Okay. Kasi yung ano yung pinipresent ko. Uh, I'll put I'll put I'll put the entire screen. All right. Now, can you guys see the clock? I'll give somebody I'll give somebody 30 seconds to answer my question. Who would want to answer my question? Hindi ko muna sasabihin yung question because there will be a prize. I'll give 30 seconds. Who wants to participate in my first game show question? Who wants to participate? Hindi ko sasabihin kung ano muna yung question. Who wants to participate? Anyone? I think you already have the idea of how I'm going to ask. You have a clue of what question I'm going to ask. But I just want people to participate. Angela is the only one who wants to participate. There's a prize, guys. There's a prize. All right, Maria Bulawin. All right, Angela and Maria Bulawin are the first people who wants to participate. What about Earl Palmero? Earl Palmero, do you want to participate? Or John Ilagan? Friendiza. All
Sige
right.
po, sir.
All right. So there's four people who want to participate. So I'm gonna give you guys 30 seconds to answer my question. All right. I'm gonna give you guys 30 seconds to answer my question. Okay. Are you ready? Are you ready? Wala ako naririnig. Oh my God, wala ako naririnig.
Ready po.
Si Angela lang. Si Angela lang ang ready.
Ready po.
Alright. Which... Oh, wait. Nasa 30 seconds. Which subclass of IgG is capable of not cross is not capable of crossing the placenta. 30 minutes on the clock. Follow the format, guys. 30 minutes on the clock. Follow the format. Answer. Unmute yourself. Unmute yourself.
Sir, namamas ko
Sir,
po.
namamas
May
ko po.
Sino na una? Sino na una sa inyong dalawa?
ako po.
Oh, wait. Stop the clock. You have 2 seconds to answer the question. Now, when I say go, you have to answer the question. Angela, is it or Angela or Friendiza?
Sige,
Angela po.
Si Angela. Okay, okay. So you have 2 seconds to answer the question, ha? Are you ready? One, two, three.
sir, namamas ko po. My name is Angela
Up!
Felipe.
Time! Time's up! Ah, sorry. But okay, let's
Okay
continue.
lang po.
But let's continue. All right, what's your answer? Namamas ko po, anong answer mo?
IgG3.
It's the one that is incapable of crossing the placenta. That is correct. All right, so email me your answer later. Your um what do you call this? Email me your globe number later. I'll send you a 50 peso GCash. All right? Globe numbers only, huh? Please. Thank you. All right, so that's how you answer the question. You only have 2 seconds left. I know you're not gonna be able to say it fast, so I gave you that one already. Okay, so two people have already TM ano to? TM po ako, sir. Pasok po 'yon. I'm not sure. I'm not sure. Naku, kawawa naman si Angela. Angela, kawawa ka naman. Gcash po. Hindi ko alam kung ano eh. Basta Gcash lang kasi usually ang inaano ko. Ikaw lang yung nagsabing TM. Meron pa bang TM sa Pilipinas? Really? Alright. Bahala ka dyan, Angela. Basta alam ko globe lang. Tingnan natin, ha? Tatry natin. Masisend ko kay mother. Si mother kasi magsisend ng ano, ng prices. Okay? So, yan. How are sensitized RBCs cleared from the blood? So basically, when we say clearing, this is the removal of blood sensitized RBCs. So RBCs that are IgM, IgG sensitized RBCs are cleared intra or extravascularly for IgG. For IgM, it is cleared intravascularly. What do I mean by this? It becomes hemolyzed immediately. All right, because of IgM. Why? IgM is a better complement fixator. While IgM, it needs to be recognized by splenic or liver phagocytes in order for it to be destroyed. All right, so that's the reason. That's what I mean by sensitized RBC clearing. All right, what do I mean by sensitization? This is the binding or fixation of immunoglobulin classes or the FAB portion of the immunoglobulin in the RBCs. For IgG, because it's monomeric, it will only bind, but it will not agglutinate. It's not as effective as for complement fixation. All right. Unlike for IgM, there are several binding sites, and then it can be easily recognized by phagocytes and it can be easily recognized by phagocytes in the bloodstream, and therefore they will be destroyed in the vascular space. Hence the term intravascular clearing. All right, for IgG sensitized RBCs, they will need to pass through the splenic arteries and the hepatic arteries, and then they will pass through the they will need to be recognized. They will be recognized as um, senescent cells or dying cells because they've already been marked. So parang minarkahan ka na ng IgG lang. Sa IgM, talagang mag-activate si complement. Kaya, at saka yung opsonization properties kasi niya, mas madaling ma-recognize ng mga phagocytes natin sa bloodstream. Alright? Now, how are IgM and IgG antibodies detected in the cross-match? Well, basically, you need to do several phases. Okay? So one is the immediate phase, the other one is the 37 degrees Celsius phase, and the other one is the AHG phase or the anti-human globulin phase. For IgM, you can immediately recognize it because these are cold reacting antibodies. They will react at 20 to 25 degrees Celsius, hence the term immediate spin phase. Okay? For AHG, for IgG, you need to do anti-human globulin phase in order for it to detect them. Now, what is the reagent that we use for AHG? Not a question, not a prized question, all right? Not a prized question, but please humor me. What is the reagent that we use for AHG? The anti-human globulin test. Somebody already answered it. I'll go and back read. Although the her question was her answer was incorrect previously. But in this question, it's correct for this question. It was actually Rachel Tesoro who answered the correct, who answered the answer to my question. What reagent do we use for what do we use for AHG? Anybody? What is the reagent that we use for AHG? Ano nga ba, sir? Anybody? Gina, are you still there? Um, who else is there? Gina, are you there? Apple, are you there? Apple Lagrana. Oh, di ba?
Sir, yung polyspecific monoclonal.
Haba naman ang sagot ni Gina. Anong
Rabbit po yung
Uh-huh. All right, I'm listening. I'm listening. What are you saying? Polyspecific monoclonal antibodies. All right.
opo.
Tapos, may sinabi ka pang extra. Ano pang sinabi mo?
Yun,
Yun lang. May sinabi ka pang rabbit eh. Ano yung rabbit na pinagsasabi mo? Sayang Gina. Ano 'yon? Sayang Gina. Sige, sino pang mag-aano? Sino pang mag-answer? Angela said Coombs. Coombs. Coombs ano? Coombs reagent. Hilig talaga ng mga students ng kulang-kulang ang sagot. Maski dun sa kabilang section na tinuturuan ko ng backtick, kulang-kulang din ang sagot. Alright, so it's Coombs reagent. Alright? Anti-human globulin can be the other term. Um, but the most common term is Coombs reagent. Why? It's from the Coombs test. What is a Coombs test, by the way, guys? There are two types of Coombs test. We have indirect and direct. Direct Coombs test detects the sensitization in vivo. Indirect Coombs test detects the sensitization in vitro. All right, so please don't confuse those terms. In the blood bank, we do what? Direct Coombs test. What do I mean by this? Because we're talking about sensitized cells. You don't need to attach the you don't need to attach the antibodies to the RBCs. All right? You don't need to attach the antibodies to the RBCs. So what do I mean by attaching the RBCs? Sir, pwede ko bang gawin yun, sir? Of course, you can do it by just the addition of antibody, the antibody reagent. But in AHD, the direct one, the moment you collect the blood sample from the patient, and we assume that the patient sample is sensitized already, meaning that there are fragments of IgG in there, specifically the FAB portion, that's the time we can use the AHG test or the AHG reagent. Okay? So that's direct Coombs test. Okay? So do you guys understand the difference between direct and indirect, or do I have to discuss it further? You can ask me now because if we go further, if you don't understand anything, that would be much more difficult for you. Do you guys understand the difference between in vitro and in vivo sensitization? Ay, walang sumasagot. Walang sumasagot. Angela, Earl, John Mediana. Is it okay? Do you understand the difference between AHG, between direct and indirect Coombs test? Oh, nobody's answering. All right, I will move on. I will assume that everybody understands it. And in the exam that I will make for you guys, I will I will ask a lot of questions about AHG. All right. Now, let's move on with a true or false questions. Hemolysis is counted as a positive result in blood bank tests. All right. I'll give you guys a minute for this one. Um, you can type it in the chat, your answers on the chat. True or false, hemolysis is counted as a positive result in the blood bank, in blood bank tests rather. 1 minute on the clock. I will just drink a cup. I will just drink water. All right. So hopefully the class will answer in a bit in consensuous. In the consensus is of a majority answer. Okay. So I'll give you guys a minute. All right. Let's check everybody's answer. All right. Let me see. Let me see. Everybody's answering. Maria, what's that? What did you answer? Polyclonal, monoclonal H3. Ay, layo. Sorry, sorry. Iba pala yun. So it started with Angela Felipe answer. The answer was true, 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 true. Everybody answered true, 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 true. All right. Let's see the correct answer. Correct answer is true. Yes. Hemolysis is actually an indication of complement activation. Why is it why is it important that we know that complement activation happens? Because cells who have been sensitized, or specifically RBCs that have been sensitized, are going to be are going to activate complements. All right. Therefore, hemolysis is an indication of a positive result. Okay. Basically mean hemolysis basically means it's an indication of positive result. All right, now let's talk about hemolysis encountered in the blood bank. There's two types of hemolysis. One is complete, one is partial, and the other one is there's no hemolysis at all. So what do we mean by this? Complete hemolysis means that there is a red supernatant and no cell button. Partial hemolysis is a red to pink supernatant with a cell button, and no hemolysis at all means there's clear supernatant with a cell button. All right. So basically, that's what we answered in the gel technique. All right. So in the gel technique, what happens? Can we say that that in the gel technique you will encounter hemolysis also in the gel technique? Um, in the gel technique, because it's been diluted. Usually we use diluents in that one. Hemolysis rarely occurs, and um, hemolysis for gel technique isn't useful. I think that's one of the disadvantages of gel techniques is that it's inconclusive if there's hemolysis, because one of the things that's important in that's important in gel technique is the grading only. The part where there is hemolysis, that's the part where that's the disadvantage of um, gel techniques. Okay, so if we're going to name one disadvantage of gel techniques, it cannot be used it cannot be used as an indicator of hemolysis of complement activation because its sole purpose is to detect the cells that are passing through the gel pillar or the microbead pillar, all right, or microbead column, right? Because antigen and antibody reactions are governed by the law of mass attractions. These types of reactions are considered to be reversible or irreversible. Irreversible. I made a typographical error. Sorry. It's supposed to be irreversible. Wait, let me end that. What's this? Oh, oops. That one is irreversible. Let me change that one and I will send another one later. It's irreversible. All right. So what happened? All right. Okay. Next, we're going to move on with the next one. Affinity versus avidity. What is affinity? This is another concept in immunology that you need to remember. Um, Initial force of attraction between a single binding site is for affinity. The sum of all the attractive forces of multivalent antigens against multivalent antibodies is avidity. So that's the difference. Usually they would ask which of the following is true regarding affinity, which of the following is true regarding avidity. So just remember there is a single binding site binding to a single there's a single binding site on an antigen, and that antibody and an antibody will and a single antibody will bind to it, whilst avidity is the sum of attractive forces between multivalent antibodies and multivalent antigens. I'll give you a great analogy of this. IgM is pentameric, true or false? Anybody in the class can say IgM is pentameric? You can tell me otherwise if I'm not correct. IgM is pentameric. True or false? True or false? IgM is pentameric. Sir, I don't know. IgM is pentameric. Anybody? True po. Okay, so everybody said true. All right. So how many binding sites are there in an IgM molecule? In a pentameric IgM molecule? A lot of people said yes, true. So there's 10 IgM binding sites, okay? One for each antibody molecule. All right? Now, they're all joined by a chain chain, and each of the binding site adds an avidity score. So basically, an avidity score is the force of attraction between a single antigen and everything. So which means that if the question is there is more avidity in a multivalent in a multivalent molecule. Yes, that's correct. So they will present these types of questions to you guys, and they are going to try to confuse you because they're going to paraphrase the questions. And you might not be able to understand it because if you may not be able to answer it correctly if you don't understand the concepts of that I'm telling you guys right now. So multivalent forces, ha. So when I say multivalent forces, multivalent antibodies, it means that there is more than one binding sites. Okay, more than one binding site rather. Okay? So if they ask you a question, which of the following has much more has a higher avidity score? Which of the following which of the following has an attractive force to a single binding site. So these are the things that you need to remember. Single for affinity, multiple for avidity. All right? Clear? All right, clear. Let's move on to the term used to denote the quality of an antibody to bind to an antigen that has caused its production. So we are going to talk about the specificity of an antibody. All right, so the ability, the, the quality of the antibody to bind to an antigen that cause its production. By the way, a review, guys. Antigen can be an immunogen, but not all antigens are immunogenic. So if an antigen is an immunogen, the process will go as follows. It will start with antigen being digested or being phagocytized by the APCs. APCs will present it to your to your lymphocyte. Specifically, which lymphocyte? A CD4 lymphocyte. CD4 lymphocyte will be sending signals to call out your other lymphocytes. What are the other lymphocytes? Your cytotoxic T cells and your helper B cells. Helper B cells will then recognize that, oh my gosh, this is an organism. This is something that is foreign, and therefore it will morph, or it will differentiate into a plasma cell. That plasma cell will then produce an antibody against the antigen that is being presented by the APC. Gets the basic summary of cell mediated response? Gets? Hopefully you get it. Hopefully you remember, and because we can move on, because this is blood banking, so we're not going to dwell deep into these things anymore. All right, let's move on to, so that's the answer to this question, specificity. That's the reason why the thing that I mentioned earlier, that the thing that I've summarized for you is how an antibody is significantly much more specific than that of your innate immunity. All right? The specificity of an antigen and antibody reaction is as such because of what concept? The lock and key concept. So the A antigen will bind to the anti-A only. All right? Unless of course there's cross reactivity, but that rarely happens again in nature. All right? So the lock and key concept. So what are the types of chemical bonds associated with antigen and antibody reactions? Well, there are four actually. If I remember correctly, one is if I'm not gonna look at the next slide. We're not gonna look at the next slide. I'm gonna try and remember electrostatic forces, hydrogen bonds, hydrophobic bonds, and the van der Waals forces. Van der Waals forces. Okay. All right. So yes, I can still remember. I'm not that old pa pala. I don't have early signs of Alzheimer's pa pala. All right, so electrostatic forces, basically this is the negative and the positive charge. Um, Anybody can tell me what is the charge of the RBC surface in normal saline solution? What is the charge? Is it a positive charge or a negative charge? What is the charge of a of an RBC in normal saline? Oh, choices. Choices. A, positive. B, negative. That is an answer. That is a question that you need to answer after I discuss each of the following types of bonds. So the electrostatic bonds is the positive and negative bonds. Hydrogen bonds are basically the bonds from which it will either be for protein molecules, okay? And then for protein molecules, the COOH and the NH groups, that's for hydrogen bonds. And hydrophobic bonds are actually very rare, very rarely discussed. But I'll give you guys an example of this. Remember your first year or second year biochemistry classes. Um, Hydrophobic bonds don't necessarily bind to water, right? They don't react to water, hence you have a lipid bilayer in your cells. And the cells can interact with each other because they have hydrophobic bonds. Water, on the other hand, will just act like a surface, like a medium for the cells to move about. So in hydrophobic bonds, what happens is that cells are attracted, the surfaces of lipid bilayers are attracted to each other as water moves away from as water molecules move away from these bonds. Okay? And van der Waals forces is basically the weakest among all the things that you're going to see here. All right? And we're going to discuss it later. What else is there? Now, there are two phases of antigen and antibody reaction. I've already mentioned it before. Sensitization is one of them. Okay, the initial binding of an antibody to the antigen binding site, which results to the deposition of complements occurring in this stage as well. All right, complements will deposit themselves as well. Lattice formation is the immune complex network formation of sensitized RBCs. Now, I have a question again. Did you guys answer already the initial question that I posted? Only two people answered. What is the charge of the RBC? Negative po, negative, negative. All right, so people answered correctly. Okay, so people answered correctly, negative charge. In normal saline. You will know later why I asked this question, why it's important to know what's the net charge of the RBC surface. Okay, the RBC surface. You'll know later why. Lattice formation is basically the formation of the immune complex sensitized RBCs. Okay, so you guys will see it as like the lace pattern. Um, if you look at your books, I didn't post it here because I might get copyrighted because we're recording this and we're posting it in YouTube. Okay, so yeah. Um, what else is there? What are the factors that affect sensitization? There are actually five factors: serum to cell ratio, incubation time, incubation temperature, pH, and the ionic strength of the medium. Let's talk about serum to cell ratio. Serum to cell ratio is basically the amount of serum versus the amount of cells in the suspension. There are three key concepts here that you need to remember: prozone, equivalence, and post zone. All right, prozone. How do we define prozone? Let's try with John Ilagan. John Ilagan, are you there?
Yes, sir. Andito po.
Taray talaga ni John Ilagan. Very active talaga. Every time na tinatanong ko siya, he will always answer and I love it. Now, what is prozone? Try your best. It doesn't matter if it's wrong. It doesn't matter if you Google it.
Ano po siya? Parang agglutinin, sir.
Agglutinin. All right, John Ilagan, thank you for trying. Let's try with another person. Um, Angela, I would have to reject your hand raising because we don't want to say we don't I don't want to be labeled as someone who has favoritism, ha? For now lang. Maria Bulawin muna tayo. All right, Angela, I hope it's okay. I hope you're not offended. Maria Bulawin.
Okay lang po.
Maria Bulawin, where
Yes,
are you?
sir.
Are you in the line, Maria Bulawin? Taray no, parang nasa radio show lang tayo, no? All right. Maria Bulawin. Maria Bulawin. All right. What do you mean by prozone? Prozone effect.
Excess antibody po.
All right, excess antibody. What about zone of equivalence?
Balance po or equal. Tama po ba, sir?
Yes. Okay, next. Post zone.
Post zone is excess antigen.
All right,
Antigen, yes po.
wonderful. You're correct in all three things. Congratulations. Unfortunately, there's no prize for that one, but thank you for participating. Charing, charang, ganon. All right, thank you so much. So serum to cell ratio. Now, what do we do in the laboratory to reach the zone of equivalence? What do we do in the laboratory to reach the zone of equivalence? Now, this one is a prized question. I will give 30 seconds in the clock 30 seconds in the clock. Starts now. Anybody can answer. Unmute yourself and then say it in the correct format. I'm timing, ha? I'm timing. There. 10 seconds left. Anybody can answer. It's an open question. There's 50 pesos load on the line. All right, nobody answered. So what we do is we do serial dilution. All right, so we do dilution. Okay, similar to what we do in the clinical chemistry department, we do dilution as well, but this time we do serial dilution. We want to reach the zone of equivalence. All right, we want to reach the zone of equivalence because we want to see the actual reaction. Because if there is any pre or post zone, post zone and pro zone and post zone phenomenon, we might get false negative reactions. All right, and we might miss out on something that is very important, especially if it's a cross-matching result or incompatible cross-match. All right, incubation time is also important in the cells. Okay, incubation temperature is again another important factor. pH is also important. Um, I'll ask a question again. At what pH would you think would be? I've said before that antigen and antibody reactions are irreversible, right? So, but in the case of pH changes, the cells can be the antigen and antibody reactions can actually be eluted. So that's the term that you can use. Elution. Elution. Hindi illusion ha. Elution. Hindi Bisaya dai ha. Elution. Ito yung ano. Ay, mali ang spelling. Elution. So at what pH do you think would elution occur? Is it an acidic pH, neutral pH, or an alkaline pH? I'll leave it as a question for the class. All right. Elution occurs at what pH? General lang. Is it in acidic, neutral, or alkaline? That is a question for you guys. I will leave it to you. I will leave it for you guys to answer later on. I'll see what is the consensus. Somebody already answered. Gina Evangelista answered. Let's move on to ionic strength. I asked earlier what is the charge of the general. Sasakalin ko to si Glyza. Glyza ha, nagagalit ako sa iyo Glyza. Sabi ko general nga lang, ayan na nga yung choices sa taas. Glyza ha. General lang. Mamaya na tayo sa specifics ha. Ikaw talaga Glyza ha. Pinagloloko mo ako. Alright, so ionic strength naman tayo. I will see the answers later in the chat room. Okay? Chat box rather. Hopefully you guys can answer that question. Um, Ionic strength, the net charge of the RBCs are basically in the saline is negative. This is because of the sialic acid present. Now let's move on to the factors affecting lattice formation. Zeta potential, and then the optimum zeta potential is one thing that occurs that is responsible for that's one of the most important thing that is responsible for lattice formation. Okay, and then optimum concentration of the antigen and antibody, and then the effects of centrifugation. I don't need to talk about centrifugation as I don't think I need to talk about centrifugation because you need to have at least 1000 1500 RPMs. That's the basic that's the minimum. It's up to 2500 actually, depending on the procedure, but usually it's above 1000 RPMs. Other than that, your results might be invalidated. Okay? So let's talk about zeta potential further. um, because of this is the repulsive forces present on the these are the repulsive forces present on the surface of a membrane. That's basically zeta potential. Okay, so the forces that I'm talking about is the zeta potential. So I'm going to go and explain it to you guys further. What's the answer for nobody answered my question. Illusion occurs at what pH? RBC illusion or
Nakilala ko ito kung paano siya dati.
ay
Tingin ko ganun pa rin siya. Wala pa yung
nanonood
nagbabago.
nanonood si
Nagsisisi.
April. April, hindi nakikinig sa lecture si April. Si April Blandes hindi na nakikinig ng lecture, nanonood ng Netflix or ng Viva, ha? April, ha? Alright. Okay, okay lang yan. Medyo nakaka-bore. I know. But this is for you guys anyway, so what else? Let's try again. Let's go with my discussion on zeta potential. Distract ako kay April. Akala ko kung sino yung nagsasalita na lalaki. Um, Close ko muna yung ano natin, presentation. And then let's talk about zeta potentials. All right? So, do you guys see paint? Bahala ka dyan, April. Basta pagka nag-exam pumasa ka, wala akong problema. Hindi ako ganun ka, hindi ako ganun ka petty. Huwag ka mag-alala, April. Basta promise mo lang sa akin, papasa ka sa exam, ha? Wala akong problema. Very chill ako, guys, kapag ka nung nagturo ako once sa Capitol, wala akong problema, wala akong pakialam kung hindi pumasok yung student. Basta pag pumasa siya, basta pag bumagsak siya, wag siya lalapit sa akin kapag ka Ganun kaya ako ka-chill, kaya tuwan-tuwa sa akin yung mga students dati. Kasi hindi ako nakikialam sa buhay nila. Because I was once like you and I didn't like it when the professors were out down on my business. So don't worry. Don't worry, April. I won't hold that against you. As long as pa-promise mo sa akin na papasa ka. All right? Next. Um this is the net charge of your RBC surface. Remember, right? We talked about it, correct? So imagine these are the negative charges, okay? And imagine nyo din na maganda yung pantay-pantay yung lines ko. Alright, guys? Sorry kung hindi pantay-pantay. And in between the surface is basically in between this empty space. Let's color it as blue para kunwari blue kasi watery ang ano. Alright, that one is basically saline solution, which is composed of water and the ion molecules from sodium and chloride. What happens is because you have sodium ions everywhere, it will bind to the negative charge. Bakit, guys? Ano ba ang charge ng ano? Ano ba ang charge ng sodium ions? General chemistry question to. What is the net charge of sodium ions? Is it positive or negative? Napakadaling question. Uh,
Positive.
right? So, because of the positive charge, right? Because of the positive charge, it will bind to the negative charge in the surface of the RBCs, right? And therefore, it will repel each other because it's already neutralized. It won't bind to that one. Another thing that you need to remember is chloride. Sorry, lagay ko dito yung ano. Lagay ko dito yung mga ions natin. N and chloride negative. All right? So, what would happen is these two molecules would actually bind with specific things. So, for example, sodium is positively charged. A lot of them will be here. They will bind to this one. More of them will bind to this. Oops. And then V. Okay. Control V. Control V. All right. So they'll bind to the surface of the RBCs, and therefore they will repel the other RBCs, thus preventing lattice formation. Why? What's the purpose of lattice formation by in the blood bank? Why do we want lattice formation in the blood bank? Because in the blood bank, if there is lattice formation, this means that the cells will be visibly indicated as whether or not you're typing it, your blood typing it. If it's not forming a lattice formation, it might cause the the medical technologists to think that, oh my gosh, walang reaction, so negative siya. So it's a big no-no to get false negative reactions. That's the reason why avoid, we want to decrease the zeta potential, because the lesser the ionic cloud is, when I say the ionic cloud, the mixture of chloride and sodium molecules in the suspension, the better the lesser the lesser the ionic cloud is, the better it is for your lattice to form. All right? So, ano naman ang pakialam ni chloride? Si chloride kasi, siya naman magbabind siya sa negative charge ng antibodies. Alright? So magbabind siya sa negative charge ni antibodies. Ay sa positive charge ni antibodies. Why? Kasi yung antibodies natin, specifically the FAB portion has has a net positive charge. So positive charge from chloride, ay negative charge from chloride will bind to the positive charge on the FAB portion, and therefore it will also cause the ionic cloud, and therefore it will cause a repelling force, thus preventing or thus delaying. It's not repelling actually in the sense na ayaw na niya dumikit. It will just delay the reaction. You guys understand? So when I talk about when the books tell you about the ionic cloud, it is basically the solution wherein which your cell suspension is being suspended, or within which the cells are being suspended. And the more ions there is, the more the longer the reaction will take. All right? It doesn't necessarily mean that it will not bind. It will mean that the cells will take longer to form the reactions will take longer to form a lattice. All right? Okay? Clear? Clear ang discussion? Clear na gets ang discussion, ha? Do I need to discuss? Do I need to elaborate further? Angela? Usually Angela is the one that's asking whether or not I will, So every time I need to elaborate further, if she didn't understand anything, I will refer to Angela as the standard. All right, Angela, is it okay for me to move on, or do I need to discuss further about the ionic cloud as well as zeta potentials and everything? Is it okay? Can I move now?
Okay, okay naman po, sir.
You understand it clearly. All right. Okay. So as long as in the exam you are able to understand the concepts, I do believe that you will be able to participate. You will be able to answer the questions. Now, let me close paint because we're going back to our discussion. All right. Going back to our discussion, entire screen. No, not entire screen. We want only the window. All right. So that is the concept of zeta potentials, and uh, it's not very much discussed in immunology because these are things that are usually discussed in the blood bank. All right. So what is the net charge of the surface of the RBC? I think everybody can answer it. It's negative. This is brought about by the sialic acid. All right. Why is zeta potential generated in the walls of the RBCs? Because sodium ions in the saline solution will bind to the surface of the RBC because of electrostatic attraction to sialic acid. That's what I'm talking about earlier. All right? All right. Now, why is it important to decrease the zeta potential in the red cell suspension? I've already answered that one as well. Okay? I've already answered that one as well. It is important because we want to decrease the time. All right? The more or decrease the distance between the cells. All right? And slow down and because of the distance, and it prevents it slows down or prevents the cross-linking of sensitized cells. So basically that's what I said earlier. All right, so what chemicals can be used as enhancement mediums to to bridge the gap between the red cells and the ionic cloud? To bridge the gap between red cells caused by the ionic cloud in the in the iodine solution, in the saline solution. So there's several things that we can talk about, that we can add to our suspension. We can and suspension and the anti-sera. We can use bovine albumin, specific bovine serum albumin, 22%. We can also use LISS. We can use polyethylene glycol, and enzymes may also be used to enhance the reactions. All right, so 22% bovine albumin enhances it because of the fact that bovine serum albumin is negatively is positive is another is a substance that has a lot of proteins which is negatively charged, and therefore it will bind to the positive charge of chloride, thus decreasing the ionic cloud in the solution. All right. LISS is different because LISS is what? It stands for what? LISS. Anybody? LIS. Hindi siya last international song syndrome, ha. Hindi siya last international song syndrome. What is LISS? Anybody? Gina.
Strength saline solution.
Huh? Sorry?
Low ionic low ionic strength saline solution.
All right. Wonderful. That is the correct answer. Thank you so much, Gina. All right.
Sir.
Okay, so that is what's the difference with it one? From the name itself, it says low ionic strength solution, low ionic strength saline solution, basic saline solution if you want to sound posh. Um, what's the difference between that normal saline and NSS, between LISS? The concentration or the molarity of the saline. Okay, so in LISS it's around 0.9, and 0.09 rather, and in NSS it's different. All right, we'll talk about it later on. Polyethylene glycol is important because it reduces the water concentration, and therefore it bridges the gap. It reduces the concentration of the water molecules, and thus decreasing the distance between the cells. All right. Next is the enzymes. Enzymes, we have three things: papain, bromelain, and physin. Okay, this enhances the reaction of some antigens in of some RBC antigens. So please don't forget those three enzymes: propain, bromelain, and physin. Okay, now let's talk about the actions of each media. So I've already told you the purpose of each. Okay, so this is basically the summary of what I've said. Bovine serum albumin neutralizes the negative charge in the RBC surface or the ionic cloud. All right. LISS lowers the concentration, has a lower concentration and of sodium and chloride ions, thus decreasing the zeta potential in the solution. All right. Polyethylene glycol concentrates antibodies in the solution by removing water, and enzymes either enhances or destroys RBC antigens. So those are the basic functions of each enhancement medium. Now, let us differentiate LISS from normal saline. LISS is, again, as I mentioned, is 0.03, not 0.09, rather. I'm sorry. I'm sorry about that one. 0.03. I was thinking of something else earlier. So it's made up of 0.03 molar sodium chloride, whilst NSS is much higher. It's 0.17 sodium chloride. All right? So clear? Clear with that one? Hopefully everybody's clear with that one. Um, because the next thing that we're going to talk about is the benefit of using LISS in blood bank procedures. The main benefit of using LISS in blood banking procedures is that it decreases the time of incubation. Normally, it's 30 minutes, but with the use of LISS or LIS, it's reduced to around 10 to 15 seconds, 15 minutes. Seconds tuloy. 10 to 15 minutes. Okay? So don't forget that one because that is usually a question that is asked in the board exam. So please don't forget it. That's the reason why I put it in bold. All right? All right. So what are the enzymes used in the blood bank? We use papain, bromelain, and phycin. Um, there's a separate discussion of it specifically for the MNSS group because they are the ones that. All right. So optimum concentration of antigen and antibody. We've already discussed these three things earlier. Just a review. Anybody? Earl, can you differentiate what is prozone, postzone, and zone of equivalence?
Prozone, sir, mas madami yung antibodies, zone of equivalence, pantay, sir. Tapos post-zone, sir, antigen yung mas madami po.
All right. Thank you. Thank you for that one. So if you get confused when you're in the board exam, sometimes we're nervous and we're nervous because this is something that is going to affect our careers in the future, blah blah blah. Sometimes students does forget about those things, little things like these. One way for you guys to remember is my mnemonic. Pro zones. Pag pro athlete ka, malaki ang katawan mo. So ano ang katawan sa Tagalog? Body. So pro zones, pros have good bodies, and therefore there is an increase in the antibody or an excess in antibody. Post zone, wag na nating alalahanin. Ang tandaan lang natin lagi si prozone. So yun yung ating mnemonic. Equivalence, alam na din natin yan, madali lang matandaan. Alright, so prozone, pro athletes. Ano ba ang meron sa pro athletes? Malaki ang mga katawan. And therefore, pag mas malaki ang katawan nila, mas maraming antibodies. O di ba? Walang kakwenta-kwenta, pero usually naaalala ng mga students. Alright, so this is basically the appearance of prozone and postzone in the exam, in the literature. Okay, this is what it looks like. Upon dilution, what would happen is you're going to get something like this, which is equivalence. If you dilute it too much, then you're going to have uh, more antigens, okay, than antibodies. All right. So this is from braincart.com. I don't take full responsibility. I don't take full credits on this specific picture. And let's move on. What false result may occur in prozone and postzone phenomenon? Is it a false positive or is it a false negative? False pass ba? False pass or false neg? Now I want everybody to at least try their hand on answering the question. All right. I'll give you guys 2 minutes because I'm going to on a bathroom break. Okay, 2 minutes. Try answering it on the chat. I'll see the answers later after I come back. Okay. answer. All right, hello. Okay, so who answered today? Renalyn, Amparo, and Angela said false negative. They are very much correct. Answer is a false negative result. On both, sir, yes, on both. You'll get a false negative results. Why? What would happen? Because the cells, because of the excess antibody, the antigens won't form a lattice. Um, for that's for prozone. Because of excess antigens, there isn't enough antibodies for lattice to form. So it needs to be in a sweet spot, and usually that sweet spot is known as the equivalence, the zone of equivalence. All right. Now, let's move on to the common blood sample in the blood bank. The common blood sample in the blood bank is basically EDTA blood that is 2 to 5% RBC suspension, that's made from EDTA. How do we make the RBC suspension? This is washed RBCs 3 times, okay, with normal saline solution. But sir, you just mentioned earlier that we don't want to use saline. You just mentioned, oops, disconnected. Disconnected again. I think I got disconnected again. Can you guys hear me? Hello?
Yes, sir.
Okay, good. Akala ko na-disconnect ako talaga. Narinig ko kasi nagtunog yung ano ko, yung USB port. Yung tung tung tung. All right. So, yeah. Um you need to be able to make a suspension. This is just washing it 3 times with NSS, and then just decanting it, and then just making it a 2 to 5% suspension, RBC suspension. Now, how do we make a RBC suspension? How do we calculate the 2 to 5% suspension? You need you need to do this formula. You need to calculate it using this formula, and the formula is amount of our washed RBCs plus divided by the total volume times 100. Okay, and what we mean by total volume is basically the amount of washed RBC plus NSS or LIS. Can I use LIS as a washing solution? Yes, you may. You may use it as a washing solution, but that is very expensive. Unless, of course, your laboratory is able to provide you with a constant supply of LISS, you can use it. But in most laboratories, we use LISS on the final volume on the final wash, or after the final wash rather. All right. Okay. Let me just correct what I just said. You can use LISS on the after the final wash. All right. So an ideal RBC suspension is described as what color? Oh, anybody can answer this question. Let's go with Gina Evangelista. Ay, taray. Gina Evangelista.
Tomato red po, sir.
Love it. I love it. But in my picture is cherry red. But yours is also correct. Cherry red or tomato red.